Okay, I think we should get started. We have a good number of people already online. And I'm sure there'll be others joining us throughout this session. But let me just start off by welcoming everybody to the first day of 4 days of student presentations that are being supported through the Peanut Innovation Lab. I want to start out by particularly thanking all the students who rather enthusiastically volunteered and agreed to give a presentation on their research projects that they're undertaking. I think as we hear from all of them, some of them are just started and some of them have been working for 1 or 2 crop seasons, so have more information to present. But we thought it was very important for all of them to give us an update on the research that they're doing. Also would like to thank all of the rest of you, the PIs, many of our co-PIs, many of their academic advisors, some of our external advisory members are all online. And we think that's very important for them as well to hear the excellent research and hopefully provide some good feedback to the students. And I'd like to start out just really thanking Kristen. She's the newest member of our management team, and we kind of gave her the task of organizing these sessions. And I think she's done a really good job of pulling them all together. And we look forward to a very full set of programs over these 4 days. I think just as a bit of background, I mean, the reason that we really wanted our students to present were several-fold. First off, it is something that we had traditionally always done in our annual meetings when we got together in person. We would feature the research projects of all students in the region or the country where we had our meeting. Of course, at the last annual meeting, we weren't able to do that extensively because it was virtual, but we had a few students. And the feedback that we got after that meeting was kind of overwhelming, that everyone wanted to hear more about what the students were doing. And so we reached out and did a survey quickly to see if there was significant availability and interest to do that. And like I said, the response was very positive. So we think that, you know, it is important for all of our students to, one, have an experience to present. But it's also important for us to hear about the research that's going on. Almost all the projects, or all the projects, are integral into the research that we're doing in our projects around in all of our countries. And so we think it's a very important component of getting updates on where we are. And I think you're going to hear a lot of very exciting research over the course of these 4 days. In pulling this together and looking at our students, I think we're rather pleased to see that we have, I think, almost at the current count, 36 graduate students participating in our projects. 26 of them are seeking master's degrees and 14 of them are seeking PhDs. They come from 9 countries and they're studying in 14 universities. All of which are in Africa, except for 3 universities here in the United States. So I think the very impressive cadre of students, and represents really, I think, one of the most important aspects of innovation labs is building that capacity for future research in our countries. So I think that's all that I want to say in kind of as an introduction. I think as you can see from the title slide, in looking at how to organize that, the sessions, we did try to organize these sessions to best fit the requirements of our students. One of the other feedback that we received from the annual meeting is that students oftentimes are the ones that have the most difficulties currently with the COVID-19 restrictions. Local transport may be lacking, or the university is not in session or in operating. And so We felt it was important to make sure that they could participate first. We realized that for some of us, we're getting up earlier than normal or staying up later than normal. But I think we all realize that being able to listen to almost 34 students over the course of 4 days without having to travel other than maybe to your office if necessary is really an opportunity that we didn't want to miss. So we will be conducting these over 4 days over the course of the next 3 weeks. Today we have about 10 students. Then next Wednesday and Thursday we will have another set, and then finally end up on August 26th on Wednesday with our final session of students. So just some simple logistics before we get started. We are recording all the sessions, and like with the annual research meeting, we'll make those available so that if you've missed or want to go back and review, and we'll also make it available for others who weren't able to join us in the sessions. We set this up more as a Zoom meeting, not a webinar type system, so we're all online. We all can turn on our audio or video if necessary, but obviously if you're not the speaker, we would prefer that you mute your microphone and turn off your video so that we can use what limited bandwidth that we have when we are Zooming with so many people online. We will, hopefully, we've asked all of our students to try to stick to about 10 minutes for their presentations. And that should leave us a good amount of time for questions and comments, feedback to the student. These could be done like we did with the annual research meeting through the chat window. That's at the bottom of your Zoom session if you want to type it in, and I'll make sure the student tries to address the questions or comments. We will be able to have you turn on your microphone and ask the question as well. We don't have a raise hand feature when we're using a meeting option in Zoom, but you can either type your request to ask a question in the chat window and I'll call on you, or we'll try to go around. We don't have a large number of participants in these sessions, so I think we'll be able to manage that at the end. But we hope that everyone will be able to ask the questions that they would like. So I think without further ado, we'd like to get started, and the first student to present today will be Velma Okaron, who is a PhD student at Makerere University in Uganda. And she's going to be speaking about developing resistance to early and late leaf spots in groundnut in Eastern and Southern Africa. So over to Velma. Velma, I think you need to unmute.
Yes, my machine has some problem. Thank you. My name is Velma Okaron. I'm presenting on resistance to early and late leaf spots in groundnut in Eastern and Southern Africa. My supervisors are Dr. Soraya Basholi from University of Georgia, USA, Dr. David Kalule from Nasari, Uganda, and Dr. Mildred Semakula, Makerere University, Uganda. Next. Next, please. Early leaf spots and late leaf spots are the most significant fungal diseases that attack groundnuts in Eastern and Southern Africa, and also tropical and subtropical regions of the world. These two diseases commonly occur together whenever groundnut is cultivated. However, their incidence and severity differ with respect to geographical location, season, as well as the cultivar. Single infection with either of the pathogen has been reported to cause yield loss of up to 50%, which is worsened by the co-infection by both pathogens. Although the use of fungicide treatment is effective, its use is still uneconomical because of the high cost of production and also environmental risk that is associated with it. Therefore, host plant resistance remains the most affordable and effective approach to manage these diseases compared to chemical and other cultural practices. The photos below shows symptoms of early leaf spots and late leaf spots. For early leaf spot, it usually occurs on the upper surface of the leaves, while the late leaf spot occurs on the lower surface of the leaves. In susceptible genotypes, these diseases cause complete defoliation of. Next, please. The basis of groundnut resistance to fungi causing these diseases has not been well investigated in Eastern Southern Africa. And this is worsened by inadequate knowledge of the population structure of the causal agents, as well as the pathogenicity of this leaf spot in Eastern Southern Africa. The effect of these and yield-related attributes has not been well documented. Although there is high resistance that has been reported in wild species, there is limited knowledge on inheritance of leaf spots among allotetraploid groundnuts and wild relative derivatives. Next. The general objective of this study is to generate genetic information and materials to support the management of leaf spot diseases of groundnuts in Eastern Southern Africa. The specific objectives include: one is to assess the genetic diversity of groundnut leaf spot pathogens in Eastern Southern Africa. Secondly, is to determine the effect of these two pathogens and their co-infection on yield and yield-related attributes of groundnuts. And lastly, is to determine the inheritance pattern of leaf spot resistance among the allotetraploid groundnut parents and the wild relative derivatives. Next. So what is the progress to date? For objective 1, dry leaf spot samples were collected from different parts of Eastern and Southern Africa region, and the samples were sent to University of Georgia. I was to travel to University of Georgia to carry out this study, but due to COVID and also travel restrictions, there has been delays. However, progress has been made after sending the infected leaves. Maricel, who is one of the students working in this project, has started carrying out fungal isolation, identification, and she's yet to do DNA extraction. So far, she has only managed to do the samples from Uganda. She's yet to do the samples from the southern region of Africa. Then, that, the samples will be sent for sequencing, and then we'll be able to do analysis and look at. Next. Next, please. Virulence and pathogenicity tests. Back, please. Thank you. Virulence and pathogenicity tests. This test is dependent on the results that will be obtained from the genetic greenhouse, and it will be done whereby after getting the results from will be selected to determine their particular seedlings of 20 groundnut genotypes. These genotypes will be selected on the basis of their resistance ratings following field screening under natural conditions. Again, these genotypes will comprise different botanical names like Virginia, Valencia, and Spanish. So after getting the most virulent pathogens from the pathogenicity test, I'll be able to continue with the objective 2 and now determine the yield loss using these most virulent pathogens. And this will be done by inoculating seedlings of 4 gram to susceptible. Next, please. Thank you. For objective 3, which is the inheritance pattern of leaf spot resistance, the crosses have already been initiated. New allotetraploids were crossed with LI, comprised of several wild species that were shipped from University of Georgia to Uganda. And the elite lines are the lines that we have here at Uganda groundnut breeding program. Next. Next, please. Thank you. After doing the crosses, 20 potential F2 hybrids were obtained. And these are examples of the crosses. For example, narrow nut 1T times IPA. Not easy to tell if they are true, and we hope to get the results soon in order to confirm if they are true hybrids. So meanwhile, advancing these F2 seeds because they were few, and later I'll be able to evaluate the hybrids under natural conditions. Next. So these are examples of F2 seeds. Looking at these seeds, as I had mentioned, they are very few for field evaluation. And it's also clear that there is variation in seed size from the pictures above. Next. Next. Thank you. So what are the challenges that have been facing? One of the challenges is the seed viability and low seed set in wild types. Remember these seeds, as I mentioned earlier, they were shipped from USA to Uganda, and maybe their viability might be related to change of environment. Secondly is the low plant stand and yield at harvest that is usually associated with the wild types. Then difficulty in harvesting wild types. Like for example, some genotypes, I could harvest only 2 or 3 pods, and these pods are small compared to the normal groundnuts. So it was really a challenge to harvest them. Then of course, the COVID-19 pandemic, for example, where travel restrictions, both locally and internationally. Locally, like in Uganda during lockdown, and also most countries had restricted travels. Also during this lockdown, we are forced to meet our supervisors virtually, still closed. Then labor, where we do this research, nursery, the institution has recommended few staff because of social distance. So labor was really a challenge. And then internet costs are high. Next. Trainings and meetings. I to be trained in gender, that is March 2020, BMS in February 2020, and the recent Peanut Innovation Lab annual meeting, which was in June 2020. Next. So what are the planned activities? I plan to do greenhouse experiment on pathogenicity tests and lyophilized vials from USA to Uganda. Noting I'll be able to travel and do work. And then final but not least, drafting thesis and papers. Thank you.
So thank you very much, Velma. I think we experienced a little bit of audio difficulty, but I think with the slides, hopefully everyone was able to get a good understanding of where you are with the project. It looks like it's good to see that progress is being made despite the COVID restrictions for sure. And yeah, I think our challenge will be to figure out when or even if we're able to provide the training here in the United States. As you can imagine, things are still very difficult at the moment for getting any kind of approvals to even start the process. But we definitely are watching that closely and hope that we can make that happen. But it's good to know that virtual meetings are happening, which is really the way we're going to have to probably do things for the foreseeable future. So the floor is open for any questions that anyone may have. Didn't get anything in the chat window, but if anybody would like to unmute, please feel free to. Don't. I guess one question from myself, Velma, I assume you can still hear me.
Yes.
The wild species, the Ipador and Batdors that you're using, what do we know about the level of resistance from those materials compared to the gnarl nut and syrah nut lines that you're crossing into? do they have more supposedly resistance to early and late leaf spot?
Based on the readings, they are reported to have high resistance for leaf spot, but we are going to screen them under field condition this season so that we can tell if their resistance is higher compared to our elite lines.
All right, good.
Yes.
Okay, well, if there's no other questions, I don't see anybody unmuting quickly or anything in the chat. So thanks a lot, Velma. Like I said, very, very exciting work, and we look forward to hopefully being able to see it in person one day. And but definitely, we look forward to hearing about the progress forward. So thank you very much. So I'd like to now move on to our second speaker, Yvonne Chapu, who is a master's student also at Makerere University in Uganda. And Yvonne's going to be speaking about high throughput phenotyping for disease and drought stress in groundnut. Yvonne, over to you. You may unmute yourself. I think we might want to not use the video for you. It seems like we're definitely having
okay, let's move to the next slide. Next. Yes, I'm Chapu Ivan. I'm going to be presenting on high throughput phenotyping for disease and drought stress selection in groundnut. I'm supervised by Dr. Cyrus Ongom Okelo, Dr. Okelo David Kalule, and Dr. Maria Balota. Next. Groundnut production is affected by both biotic and abiotic constraints, of which late leaf spot, groundnut rosette disease are the most important diseases, and drought is the most important abiotic factor in Uganda. Farmers will benefit from growing high-yielding, disease-resistant and drought-tolerant varieties. As you all know, most smallholder farmers have limited access to pesticides and irrigation to avert these constraints, and therefore development of cultivars with improved resistance and disease tolerance would greatly improve yield stability and quality, but the breeding process requires faster and accurate phenotyping. Next. Currently, traditional phenotyping methods are used to aid selections, but we know these traditional phenotyping methods are slow, costly, destructive, and the data collected is usually subjective. For example, if you are visually scoring disease and wilting with by different individuals in the field, you'll find that this data might usually have high variation because everyone is using his own opinion. And therefore, the quality of the phenotypic data is highly compromised. So high throughput phenotyping is needed for faster and accurate phenotyping in order to improve the breeding process and accelerate genetic gains. We all know high throughput phenotyping will reduce the phenotyping costs. We shall get objective measurements, and it's faster. These high throughput phenotyping tools have successfully been applied in wheat and maize breeding programs for drought selection and disease. However, they are yet to be tested for effective deployment in groundnut breeding or routine selection for disease and drought tolerance. And this brings me to my objectives. Next. My general objective is to develop, is to contribute to the development and deployment of HTP models for drought and disease selection in groundnut breeding. The specific objectives: 1, to determine the effectiveness of using HTP parameters in identifying drought, GRD, and LLS symptoms in groundnut. 2, to determine the effectiveness of using HTP parameters in predicting yield under GRD, LLS, and drought conditions. Hypothesis: 1. HTP models are more accurate in determining biotic and abiotic stresses compared to traditional methods. And two, HTP-based models can effectively predict end-of-season pod yield. Next. For objective one, the materials and methods, I have two populations. The first population is my training population, which is comprised of 50 commercial and breeding lanes from the nursery and equine breeding programs. This population has been planted in two locations, Nursery and Nakabango, which are GRD and LLS hotspots. And a validation population of 200 genotypes from the African co-collection, which have also been planted at Nursery and Nakabango. Data has been collected using both the visual and HTP sensors. So some of the visual traits we have collected, plant vigor, we are using a scale of 1 to 5. LLS severity, we are using a scale of 1 to 9, as described by Subramanyam. And GRD, we are using a scale of 1 to 5, as described by Naidu et al. And then for the HTP sensors, next. We are using the green seeker for NDVI, thermal camera for canopy temperature, and RGB camera for RGB indices. So the green seeker measures the NDVI, and NDVI values range from 0 to 1, with higher values being preferred because they represent healthier plants. And for the RGB camera, we use the BreedPrix software to develop RGB indices. These RGB indices are described according to the International Commission of Illumination. So we have indices like hue, saturation, lightness, A, B. Then we have the green area, which comprises of pixels for between 60 and 120, and the greener area, which has pixels between 80 and 120. Then we have the crop senescence index, which is just a function of green area and greener area. Next. So from some of the results we got, for plant vigor, we have correlation coefficients from a Pearson correlation matrix between the visually scored plant vigor, NDVI, and RGB derived indices. So we saw that NDVI and NDVI green area and greener area were best correlated with visually scored vigor. So from this, it simply shows that plant vigor is related with presence of green biomass. The higher the biomass, the greater the plant vigor. And NDVI is best at doing this because it's explaining 71% of the variation within our population, as you can see from figure 2. Next. So for late leaf spot, we also had correlation coefficients from the Pearson correlation matrix between the visually scored LLS symptoms 1 to 9, the NDVI, RGB indices, and canopy temperature. So from here, we see the crop senescence index, NDVI, and green area best estimated the leaf spot symptoms, but the crop senescence index and NDVI were the best. So the crop senescence index shows how much percentage of the canopy is dying off, and this is very evident with the late leaf spot. You have leaves dying off and falling off, and NDVI picks this very well. The increase in disease severity causes greatly reduces the NDVI because the number of reflective surfaces is decreasing. So to the in figure 3, this is one of the lines from Niger among the African co-collection. At the top, we have the plant at 6 weeks, and then at the bottom, we have the plant at 12 weeks. So you can see the effect of late leaf spot. Almost half of the plant canopy has dropped off because of the senescence. And you see the green area greatly reducing. At 6 weeks, we have a greener area of about 0.2. At 12 weeks, we have a greener area of about 0.13, and this effect is due to the leaf drop caused by the LLS. Next. For groundnut rosette disease, we did the same. We had a Pearson correlation matrix between the visual scored GRD symptoms 1 to 5, the percentage disease incidence, with the HTP parameters. So the correlations were not all that great with the GRD. We had low correlations, but still let's just look at the signs. With the green area is reduced by the increase in disease severity, and so is the greener area. The NDVI index as well reduces with increase in the disease severity. You see from figure 4, the plant is greatly stunted because of the GRD, and by stunting, the greener area is greatly reduced. And so these are some of the symptoms we can be able to quantify in the field. Next. So for drought, I was not able to carry out drought in the field because we always have rain. Rain is always falling, so there's no time for plants to be stressed by drought. So this is why we improvised a greenhouse. So within this greenhouse, we have well irrigated, and we are going to have irrigated and stressed pots. And because of the high temperatures, we are also taking this opportunity to screen these genotypes for heat tolerance, because the temperatures are really high. And sometimes we have temperatures going up to around 58 degrees Celsius. So from within this experiment, we want to see if we can quantify the leaf wilting, which we always observe in the field, because leaf wilting is one of the earliest symptoms of drought stress. So if we can be able to quantify the leaf wilting within the genotypes, that will be really good. Next. So for my second objective, determine the effectiveness of using HTP parameters in predicting yield under GRD, LLS, and drought conditions. Still, I'm using the same populations, the training population and the validation population, and I'm collecting data weekly from 4 weeks up to 12 weeks. And right now we have 12 weeks data. So we are waiting for the end of season yield, from which we can determine which parameters are best related to yield, and if we can use these parameters to predict the end of season yield. We also want to be able to understand, to be able to tell at what time we can best predict this end of season yield. Yes, next. So far the meetings I've attended under the project, we had a progress meeting with Dr. Dave Hoisington at Makerere University. This was in Feb. We had a gender training with the great team of Makerere, with together with Jessica at Makerere. Then attended the Innovation Lab annual general meeting, attended the A-Press. I participated in the graduate student poster competition. Next. So the challenges so far, the university is still closed, so we don't know what time we are going to be able to finish. The COVID, the measures are making work difficult, like weeding. We have less people working on the field, and my work is sensitive to weeds, so I always have to make sure the field is clean enough. So this always affects the time of data collection, because people are taking longer to do the weeding. And data collection off station is really difficult. We have to work within a given range of time. No night travels are accepted, so we work within daytime, and at times we fail to finish the data collection. And the online meetings have really not been that bad. At least I'm having monthly meetings with my supervisors, Dr. Maria, Dr. Cyrus, and Dr. Kello. So at least we have monthly meetings, but the biggest problem is the network. You can't really communicate in some parts of the country. The network is really bad. Next. So for all this, I would like to thank my supervisors, Dr. Cyrus Okello, Dr. David Kalwe, and Dr. Maria for how far they've brought me, and my fellow students, Esther Achola, Danielle Esando, Tabitha Lomotey, Cecilia, for all the assistance they've given me, the technicians at Nasari for helping our work move forward, and the Innovation Lab for funding the studies. Thank you.
Thank you very much, Yvonne. Very nice presentation. Floor is now open for any questions. Didn't see any come through the chat so far, but does any of our participants have any questions for Yvonne? Or any comments? I have one question, Yvonne, for you. I think we discussed it briefly when I was there earlier in the year, but I was just wondering, what are you finding out in terms of the sensitivity or the variation using the high throughput methods, the sensors, in terms of, is it varying a lot depending upon when during the day you take the measurements or which day of the week you take? And also, does it make a difference who takes the measurements, whether it's you or a technician, or how much, I guess, getting some idea of just how much variation there is?
So for this season, mostly I have been using the technicians to help me collect the data. And for the camera, RGB cameras, is where I get a bit of variation because of the height. It depends on the height at which you hold the camera. So different people with different heights will always have some small variation, but with the NDVI, there's almost no variation because, can I say, we almost hold it at a constant at about 60 degrees from the plant canopy, so there's no much variation. So there's a question in the chats.
Yeah, so Maria's asking if you think that using a combination of indices, you'll get a better estimate of any of those traits like rosette or late leaf spot or drought.
Okay, yeah, I think if, because what I have just been able to do, this has been simple linear regression, that's what I've been doing, but I'm sure if we carried out multiple linear regression using different indices, combining the canopy temperature, the RGB indices and the NDVI together, I'm sure we can get higher estimations of the LLS and GRD. And besides, the data I'm presenting is from one location. I've not been able to clean out the data from the second location, but I'm sure if we combine the data from both locations, I'm sure we can get very good variation and very good estimates.
Good. One other question from me. I'm assuming that you're accumulating a lot of data, especially from the images. How are you storing those, and are there any challenges or issues that we ought to be aware of that we could help with in terms of just making that information available in the future?
So for now, the images are I really have lots of images, and at least I have an external drive from Dr. Okelo Kalule where I store the images. And once I finish analyzing the data, we upload it to the cloud, the BMS server. Esther does that for us. So at least the data is stored on the server.
Okay, that's good to know. Guess we had a question from Joseph wanting to know whether the camera, and I don't know whether that's the NDVI one, or automatically adapts to environmental conditions.
Okay, automatically adjust. Okay. Oh, this one is quite good. I do not get the question quite clearly, but from what I can see, I think the camera doesn't need any adaptation. The only slight difference I see with the camera, it also depends on the kind of soils, because I realized, and I think the soils have to do with the environment. I realized from one location in Nasari, the soils are a little darker, in Nakabango, the soils are a little red. So when you are using you find that the A index from the RGB cameras, which represents the green or red, you find the values being more red and less of the green. And that is one thing I noticed. So I think you have to be careful with the kind of soils you're dealing with. If the soils are dark, you have to pay closer attention to the A and U indices of the RGB camera. So this is something small I noticed with the data I've been handling.
Okay.
Yes.
Thanks very much, Yvonne, for a very nice presentation, and good luck. And we look forward to hearing more about the excellent work that you're doing.
Thank you.
So we'd now like to move on to our third presentation this morning. And that's going to be given by Nogaye Ndiaye from Senegal. She is a master's student at the University of Thies. And I believe her presentation, the PowerPoint is in English, but I believe she may be speaking in French. We did offer our students the option, if they felt a little bit more comfortable in their native language, to do that in this session, since it was pretty much at the last moment. So hopefully everyone will be able to get the gist of by reading the slides, but we do look forward to your presentation, Ngoye.
Bonjour, mesdames et messieurs. Je me nomme Nogaye Ndiaye, ingénieur pédologue à l'ENSA de Thies. Je suis honorée d'être là aujourd'hui pour faire l'évaluation de la première année du projet Optimize Shrub System, dans lequel j'ai travaillé sur le thème de effet du système optimisé à Nguera sénégalensis sur des indicateurs de la qualité du sol et la performance agrophysiologique de l'arachide. Next, please. Pour mener à bien cette présentation, nous allons d'abord parler de l'introduction et ressortir la problématique et les objectifs de l'étude, la méthodologie qu'on a adoptée, les résultats premiers, pour ensuite terminer avec une conclusion et des recommandations. Next, please. OK. L'agriculture constitue la base de l'alimentation et la principale source de revenus des pays du Sahel. Malgré tous ses atouts, elle est confrontée à des contraintes telles que les aléas climatiques, la pression démographique et la dégradation des ressources naturelles. Ce sont ces diverses contraintes qui sont responsables aujourd'hui de la diminution de la production ainsi que de la baisse de la fertilité, entraînant la pauvreté et l'insécurité alimentaire. Next. Next, please. OK. L'agriculture au Sénégal, plus particulièrement dans le bassin arachidier, se caractérise comme étant une agriculture de type familial qui repose essentiellement sur des cultures de mil et d'arachide. C'est une agriculture qui dépend de la pluviométrie et qui est faiblement mécanisée. Aujourd'hui, on assiste à la disparition de la jachère, à l'exportation des éléments minéraux après prélèvement de la culture, au phénomène d'érosion hydrique et éolienne, les pratiques de brûlis et aussi une mauvaise gestion des engrais. Ceci a entraîné a fait que les sols aujourd'hui sont vulnérables et qui entraîne une diminution de la production. Et les technologies de la révolution verte qui sont aujourd'hui la solution pour résoudre cette situation ne sont pas du tout pris en compte par les paysans. Next, please. OK. Des études ont été menées en station dans le bassin arachidier du Sénégal depuis plus de 14 ans par des chercheurs français et anglais. Et à l'issue de leurs résultats, ils ont montré que les arbustes autochtones, en association avec les cultures telles que le mil et l'arachide, pouvaient impacter sur la qualité du sol et l'état hydrique du sol et aussi les rendements des cultures. Toutefois, il faut remarquer que ces résultats n'ont pas été validés en milieu paysan. C'est dans ce contexte que l'ENSA, en collaboration avec l'Université de Ohio, ont bénéficié d'un financement du programme Peanut Innovation Lab de l'USAID pour exécuter ce projet. Next. OK, l'objectif global de cette étude est de contribuer à la validation du Optimal Shrub System afin d'assurer sa diffusion large et aussi de contribuer à l'insécurité alimentaire dans les pays du Sahel. Comme objectif spécifique, next, il s'agit de déterminer en fait l'effet du OSS sur les propriétés physiques, chimiques et biologiques du sol, de connaître l'influence du OSS sur la croissance et la physiologie de l'arachide, mais également aussi de connaître l'influence du OSS sur le rendement de la culture pour la première année. Next. Notre étude a été faite dans la zone agroécologie nord du bassin arachidier durant la saison de culture 2019. Next. Et 6 villages ont été sélectionnés et dans ces 6 villages, 15 champs ont été suivis. Next. OK, pour le dispositif expérimental qu'on a mis en place. Next. C'est un dispositif à bloc complet randomisé avec 2 traitements. Il y a le système traditionnel des arbustes et le système optimisé des arbustes, c'est-à-dire le OSS. Et les parcelles étaient au nombre de 15. Donc, on a eu 15 répétitions. Et les parcelles avaient des superficies de 500 mètres carrés. Next. Next, please. Next. Next, please. Next. OK. Voici comment les paysans font pour l'arbuste. C'est-à-dire que eux, au moment de la préparation du champ, ils coupent les arbustes, puis ils les brûlent. Durant la saison de culture, ils reviennent de nouveau pour couper les arbustes, mais cette fois-ci, la biomasse qu'ils obtiennent n'est pas brûlée ni épandue, mais il est mis en tas. Next, please. OK. Pour le OSS, pour le Shrub Optimized System, ça consiste d'abord à produire des plants par marcottage. Ensuite, ces plants sont répliqués au champ par les paysans dans le but d'obtenir 60 arbustes par 500 mètres carrés. Et pour le mode de gestion, next, il s'agit de couper ces arbustes. Après coupe, le résidu qui est obtenu est épandu et incorporé dans le sol de façon homogène. Next, please. Next. OK. Pour la conduite de la culture, la variété d'arachide qu'on a utilisée dans les 2 traitements, c'est le 55437 et l'engrais NPK 62010 a été également utilisé à une dose de 150 kg à l'hectare. Et pour la biomasse, c'est-à-dire que les résidus qu'on a obtenus après coupe a été épandus de façon homogène dans le champ. Vu qu'on était en première année et qu'il nous fallait ajouter plus d'amendements, donc on a ajouté de la biomasse supplémentaire dans les champs aux environs de 3 kg à l'hectare. Next, please. Next. Next, please. OK. Voici les images qui illustrent en fait comment l'arbuste est valorisé pour la régénération du sol. Next, please. Next. OK. Des mesures pour la méthodologie, des mesures ont été effectuées sur le sol. L'échantillonnage s'est fait sur 2 périodes, avant la mise en place de la culture et après la récolte à 2 profondeurs. Donc, pour cela, les parcelles ont été scindées en 3 sections. Et comme vous le voyez, dans chaque section, 15 points ont été échantillonnés puis mélangés. Au laboratoire, on a effectué des analyses chimiques, à savoir le carbone, l'azote et aussi le phosphore. Next, please. OK. Pour le stock hydrique du sol, c'est-à-dire que l'humidité du sol a été mesurée à l'aide de la sonde hydroxense 2 à 2 profondeurs, 12 cm et 20 cm dans les parcelles élémentaires. Next. Des mesures également ont été faites sur les cultures, à savoir, on a fait une observation phénologique, puis on a mesuré certains paramètres de croissance et aussi on a mesuré des paramètres physiologiques. Après, à la fin de la campagne, on a procédé par l'évaluation du rendement. De ce fait, le poids de 100 graines, le rendement en gousse a été également mesuré, déterminé. Next, please. OK, voici les résultats qu'on a obtenus à l'état initial des sols. Next. On a constaté que les sols étaient de type sable après analyse au laboratoire et que également les pourcentages de limon et d'argile étaient très faibles dans les 2 parcelles. De même que les teneurs en éléments chimiques étaient également très faibles dans les 2 parcelles. Et aussi, les sols étudiés possédaient également un pH acide. Ça, ce sont les caractéristiques en fait de l'état initial du sol. Next, please. Next. OK, cette figure-ci montre l'effet des traitements en fait sur les teneurs en carbone du sol. D'après l'analyse de variance qu'on a effectuée, il y a eu une différence significative du taux de carbone. De ce fait, on remarque que le PI, c'est-à-dire le Optimal Shrub System, avait des valeurs beaucoup plus importantes que par rapport au système traditionnel. Next, please. Également, ceci représente l'effet des traitements sur les teneurs en azote. Nous avons remarqué qu'il n'y a pas eu un effet significatif, mais malgré qu'il n'y a pas eu un effet significatif, next, on a pu constater que le traitement PI présentait des taux significativement supérieurs dans les deux parcelles par rapport au système PP à 2 profondeurs différentes. Next, please. Next. OK, pour les paramètres agronomiques, on a la conductance stomatique a révélé l'absence de différence significative. Tout de même, on a remarqué que le PI avait des valeurs beaucoup plus importantes par rapport au PP. Et pour les mesures phénologiques, à savoir la date de levée, de 50% de floraison et de 50% de maturation, les dates étaient les mêmes dans les 2 traitements qu'on a étudiés. Next, please. OK, cette figure-ci représente le taux de survie des arbustes en fin d'hivernage. Next, il ressort de cette graphique qu'on avait planté 461 arbustes au départ. Et en fin de campagne, on a évalué et qu'on a trouvé 382 plants. Donc, on a eu un taux de survie de 82,86%. Next, please. Next. OK. Pour les paramètres de croissance, l'analyse a montré qu'on n'a pas eu un effet significatif. Mais tout de même, next, on voit que les valeurs maximales étaient concentrées dans les traitements PI. Next. OK, que les valeurs étaient beaucoup plus importantes dans le traitement PI par rapport au traitement PP. Next, please. C'est le next. C'est le même constat fait pour la biomasse produite. C'est le même constat fait pour la biomasse produite. Il n'y a pas eu une différence significative, mais le traitement PI présentait également des valeurs beaucoup plus importantes par rapport au PP. Next, please. Cette figure-ci représente en fait l'effet des traitements sur le rendement en gousse. Il n'y a pas eu un effet significatif, mais les résultats montrent également que le traitement PI présentait en fait des taux vraiment plus importants par rapport au traitement PP. Next, please. OK. En conclusion, nous pouvons dire que l'objectif a été de comparer les performances du sol et de l'arachide pour la première année de l'établissement du OSS. Bien qu'il n'y ait pas eu une différence statistiquement significative sur les paramètres physiologiques et de rendement, une tendance d'augmentation a été constatée dans les traitements PI par rapport au système traditionnel. Et aussi, il faut noter que pour avoir une réponse significative, il faut attendre 2 à 3 ans pour que les arbustes nouvellement plantés puissent s'établir, car les arbustes ont été plantés tardivement en raison de financement et d'autres facteurs. Et aussi, on a remarqué que le déficit hydrique qui est dû à une faible précipitation, le retard de semis ainsi que le type de sol ont fortement influencé sur le rendement de l'arachide. Également, l'OSS a pu montrer une augmentation du taux de carbone dans le sol, ce qui laisse supposer que l'OSS améliore en fait la qualité du sol tout en séquestrant le carbone pour la première année. Et également, on a pu constater que le taux d'azote était plus important dans le OSS. Et aussi, pour le taux de survie, on a constaté qu'il y avait un taux de survie de 82% pour les plants nouvellement plantés, et ceci est conforme avec les recherches antérieures. Next, please. Next. OK. À l'issue des résultats obtenus et aussi les limites de l'étude, quelques recommandations sont reformulées. En fait, il faut introduire plus d'amendements d'Nguera dans les parcelles pour ressortir en fait ces effets bénéfiques par rapport à la qualité du sol et le rendement. Mais également, il faut semer très tôt afin d'éviter les retards de semis, de maintenir les entretiens fréquents dans les champs, mais surtout de sensibiliser les paysans par rapport à la réalisation de pépinières d'Nguera et aussi d'impliquer plus de partenaires dans le projet pour une diffusion plus large. Next, please. Next. Voici le calendrier que nous proposons pour l'année 2020 pour la suite du projet. Next. OK, ceci met fin à la présentation. C'était ça, l'évaluation qu'on a fait de la première année. Nous attendons les questions pour une meilleure compréhension. Thank you for your kind attention.
Thank you very much, Nogaye. Very nice presentation. I think everyone probably were able to get quite a bit from it. The floor is open for any quick question if anyone has it, maybe while you're unmuting and all. I would just ask you one question. What is the peanut variety that you are using in the fields?
OK, on a utilisé la variété 55437. C'est une variété qui est tolérante aux 400, yeah. On a utilisé en fait la variété 55437. c'est une variété qu'on utilise dans le nord du bassin arachidier.
Okay, thank you. Good. Any other questions? Cynthia would like to know whether this year has been typical and what is the survival rate of the plants that you would expect?
Ok, d'accord. Les difficultés qu'on a rencontrées par rapport à l'étude, d'abord avant l'établissement des essais, parce qu'on avait eu un retard de semis. Et donc, cela a impacté vraiment sur le rendement des cultures. Et aussi, au cours de l'étude, on a eu des problèmes de financement. Donc, du coup, on avait des difficultés en fait pour déplacer et aller prendre des mesures. Et par rapport à ce que nous attendons, on savait que comme c'était la première année, c'était pas du tout évident d'avoir une réponse significative. Donc, peut-être les années qui vont suivre, on va avoir plus de rendement et aussi on aura un niveau de fertilité beaucoup plus important. Thank you.
Okay, we also have a question from Jessica, which if you can read the chat, you can read it in French, or it's, what do you think are the obstacles to adoption of this optimized shrub system in Senegal?
Les obstacles à l'adoption étendue au système admis par les agriculteurs. En fait, les obstacles pour épandre ces arbustes, c'est-à-dire que le fait de découper les arbustes et tout ça, c'est quelque chose de très difficile chez les paysans parce que ils aimeraient avoir une machine qui va s'en charger de découper au lieu de le faire manuellement. Donc, c'est la difficulté que les paysans ont pour faire ce travail. Et vraiment, ils sollicitent à avoir des machines beaucoup plus performantes qui vont découper ces arbustes donc pour une utilisation beaucoup plus facile. Et aussi, il a parlé des hommes ou des femmes. Ben, c'est les deux, en fait. Ce sont les hommes, ce sont les femmes qui font ça, les deux. Donc, il n'y a pas une distinction de genre dans le travail. Thank you.
Okay, thank you. I think we'll need to move on. Merci beaucoup. Nogaye?
Okay, thank you.
Good luck.
Okay, merci.
So we'd like to move on now to our next presentation, which will be by Precious Mtengezo. Precious is a master's student at Lilongwe in Lilongwe, Malawi. And he will be speaking about the responses of groundnut varieties to rhizobia inoculation, fertilizer, and plant density. Precious, we look forward to your presentation.
Hello. Hope you can hear me clearly.
Yes, you're breaking up just a little bit, but I think it'll hopefully be okay.
Yeah. My name is Precious Mtengezo. I'm a master's student in agronomy at Lilongwe University of Agriculture and Natural Resources. My project title is Response of Groundnut Varieties to Rhizobia Inoculation in Organic Fertilizer Application and Plant Density. My work is being supervised by Dr. Muhango and Dr. Brandenberg, plus other two supervisors here at Rwanda. Next slide, please. Next. Hello. Yeah, groundnuts is one of the oilseed legume crops grown for food and cash. For here in Malawi, it is one of the crops that is mainly being encouraged by the government. It was also stipulated in the National Agriculture Policy of 2016 that oilseed crops like groundnuts should be used as a substitute for tobacco production. And in Malawi, the major two varieties that are being used is Virginia and the Spanish. But the major issue in Malawi is there's low productivity of groundnuts, mainly for smallholder farmers, of yields ranging from 500 to 800 and even lower, below 500. Yeah, but the major contribution to the low yields in Malawi is due to low soil fertility and the plant densities and also, but also the diseases and the poor varieties that are being used in Malawi. The major issue for this is lack of investment in inputs in agriculture production. Many farmers with this thing that groundnuts are able to fix nitrogen on their own, usually they don't apply much inputs in groundnuts. Next, okay. A lot of studies have been done. Mainly, they are targeting at finding influence of different fertilizers on groundnuts, but mostly they consider a minimal number of factors like how do phosphorus fertilizer affect the productivity of groundnuts. But this trial is combining a lot of factors to see their effect on groundnut production. Next. Yeah, the objective of this trial mainly is to evaluate how application of decomposed fertilizer and seed integration and also varying different plant densities will affect BNF and plant yield of groundnuts. And those are the specific objectives as they have been separated. Next, for the hypothesis, the first hypothesis is that application of decomposed and rhizobia in seed integration to groundnuts at high plant density will increase BNF. And for the second one is that application of decomposed fertilizers and rhizobia seed integration will increase yield and yield components of these groundnut varieties at a high density. Next, please. These trials are being done in Lilongwe, that's central Malawi, 2 sites, Thies site and Horizon site. Next, for the characteristics, so the soil, the baseline soil characteristics, those are the ones that have been shown in table 1. Mainly, on pH, we can see they are slightly acidic to near neutral soils, which is actually providing an ideal soil, although slight acidity may affect the productivity of groundnuts, but the other parameters are there as shown in the slide. Next. Yeah, for the trials of this, for the treatment of this trial, there were four factors, and each one with 2 levels, whereby we had a plant density where there's low and high levels, but the high level was attained by doubling the plant flows and the varieties as shown there, and in organic fertilizer application, decomposed was used, and in other treatments, it was applied where in others it wasn't. And the seed inoculation, and all these factors in that order they have been treated, they were splitted, and there were 4 lubricants. Next. Yeah, the data that were being collected are shown in that slide, the soil data and crop data as it has been listed there. Up to last BNF was estimated. BNF main range was estimated using the modified nitrogen difference method adopted from GIRA 2001. Next. Some of the data that was collected, the results are shown there. We have days to 50% flowering and days to begging. There's a significant difference mainly due to varietal effect, and usually that is genetical. Next. Yeah. On table 4A, there is a results on soil percentage and groundnut grain yield. That's, I've separated them based on the site that I was conducting the experiments. For the grain yield, there are significant differences on starting with plant density and variety, but also the fertilizer application. That is, for the plant density, I think that is already expected that at higher densities we are having a higher yield than at lower densities. But on variety is also genetical. While on the fertilizer application, G compound shows that it has increased the yield of groundnuts as compared to those that were not applied. Next. In the previous slide on the yield, there was on the grain yield, there was an interaction on variety and plant density. So this slide is just summarizing how the interaction is happening. We can see that CG9 is performing better on low density than at higher densities, while Thies is performing better at higher density than on lower density. CG9 is a Virginia variety, so it requires a lot of plant nutrients for it to complete its life cycle. So we see increasing the plant density in this case affected the productivity of CG9, which was maybe unexpected as according to the hypothesis. Next. Next. Okay. Yeah, that's the results on the dry pod, sharing percentage, and grain yield and a BNF for today's site. Mainly for sharing percentage, it's a varietal effect whereby Thies is doing better in terms of percentage of real grain to the total dry pod yield. While on grain yield, there is a significant difference in terms of from varieties and to fertilizer application, but nothing according to inoculant application. And in terms of BNF, it's a CG9 that is topping up, that is at a higher levels of fixing nitrogen than the Thies variety. Next. That's that table. That figure is just showing the nitrogen that has been fixed. That's according to horizon site. As you can see in the table, the factors, there are 4 factors: the plant density, the variety, the fertilizer application, and the inoculant application. They are acting as separate factors, meaning that the error bars from plant density are just focused on plant density, and the other factor goes. And from this figure, there is a significant difference on amount of nitrogen that have been fixed. That's nitrogen benefit from varieties where CG9 is performing better. CG9 is a Virginia, while Chidara is the Spanish variety. So the Virginia type is performing better than the Spanish variety. But also in terms of biological nitrogen amount of any fixed according to fertilizer application, those trials those treatments that received decompound fertilizer did better, unlike those that didn't receive. But there was no significant difference in terms of inoculant application and plant density. Next. In concluding, application of decompound fertilizer increased both grain and BNF. CG9 fixed more than Chidara variety. Well, in terms of plant density and inoculant application, the BNF, there was no significant difference in there on how they fixed N. On challenges, there was a delay in planting according due to some logistics, but it had some effects on the yield, but also poor seed germination mainly for CG9. So this was a thing to do with seed quality. Yeah, apart from this, the other challenges include the COVID-19 pandemic that we have been hit, but mainly according to the trial itself, those are the challenges that I faced. On way forward, I'll be continuing with my data analysis to other treatments to other data parameters that I collected. Though they have not been presented, but there's a bag of them that maybe if opportunity arises, they will be presented and they will also be included in the reports. But also, this trial will be also done in 2020-2021 growing season. Next. Acknowledgements. Thanks to Feed the Future, the USAID, and the Peanut Innovations Lab from Georgia for the support for the funding of this project. And thank you all for your attention.
Thank you very much, Precious. Good presentation. The
Thank you.
floor is open to any questions for Precious. I had one question, maybe I missed it in your presentation. What was the varieties that you used for the different treatments, such as plant density and fertilizer and inoculant?
I used two varieties. That is CG9, which is a Virginia variety, and Chidara, which is a Spanish type variety.
So the data that you presented then for those treatments is the average of those two varieties, or did you see any differences between how those two different varieties reacted to each of those treatments?
Yeah. Mainly in terms of yield, you see that the grain yield itself, the Chidara did better mainly out of the horizon farm than the CG9. We always know that Virginia does better than, or even from their potential yields, Virginia does better than the Spanish. But in this case, maybe due to the late planting, Spanish did better than the Virginia. But in terms of their ability to fix N, we see that CG9 fixes more N. So as you already know, the amount of N fixed is so dependent on all the total plant biomass. So CG9 did better in terms of ability to fix N. But for venue, Zahra did better.
Okay, thank you.
Yeah, thank you.
Any other questions for Precious this time? Okay, well, thank you very much, Precious, and we look forward to hearing more results as you get them, and good luck for the coming year.
Thank you very much.
So we'll now move on to our next presenter, which is Stephen Arthur. Stephen is a PhD candidate at KNUST University in Ghana. And Stephen will be talking about the effects of pest and crop management packages in groundnut, looking at cropping systems on agricultural productivity. Over to you, Stephen.
Okay, thank you. Thank you, Dave. So I'm Stephen Arthur, and my work is on effect of pest and crop management packages in groundnut-based cropping systems on agricultural productivity. And I'm doing my schooling in Commey University of Science and Technology in Ghana. And this is my graduate committee. Next slide. So this will be the outline of my presentation. I'll take you through the introduction and then the materials and methods and a few results that I've been seeing, and then my work plan. Next. So the importance of peanut cannot be overemphasized to a group like this, but apart from its use as food and feed, it plays a lot of other important roles in our communities. Talk about marriages and naming ceremonies and others. They play major roles in there. They are also very important on our field crops and predominant among the benefits that we have are the abilities to fix atmospheric nitrogen and make them available to the next crop, and also their importance in management of weeds, as well as its use as a component in insect pest management, especially in the infamous push-pull system for fall armyworm control. So in Ghana, peanut is cultivated by peasants. Next slide. And it's usually done, cultivated as a sole crop or as an intercrop with cereals or cotton, or in rotation with cereals, cotton, or root and tuber crops. Yields remain lower, and yields of about 76% lower has been recorded as against the potential minimum yield of 2.5 tons per hectare. And this shortfall can be attributed to a lot of things, but prominent amongst them would be poor seeds and varieties that are susceptible to pests and diseases, planting at time that is late, and poor soil nutrition, as well as inappropriate weed and other pest management and disease management. Next slide. So a lot of research work has actually gone on, and of research work has gone on, and recommendations have been given on individual basis, and farmers have not really treated these as systems so as to get much from the recommendations. And research is partly to be blamed for this, because research has not actually given farmers enough reasons to adopt these recommendations as a system. And this is because the long-term benefits to soil nutrition and crop, yield improvement, as well as management of weeds and other pest management has not been really demonstrated to farmers. And also the extended cost and time benefits analysis and the benefits that farmers will get with this has also not been proven. Next slide. And so it is believed that with good agronomic practices, the components in there would complement each other and would result in improving agricultural productivity. And so this work was set out to actually develop sustainable medium to long-term groundnut cropping systems for enhanced adoption of agricultural technologies. Next slide, please. So specifically, I'm working on 4 objectives, and the first is to study the dynamics of pest and disease incidence in peanut cropping systems, and also to look at the dynamics of soil quality properties in peanut-based cropping systems. With this, soil samples are being collected at the beginning of every season or cropping season, and these will be analyzed to look at the dynamics. I would also look at the evaluation of the effect of cropping and management systems on crop productivity and quality, and then determine the economic viability of the peanut-based cropping systems. Next slide. So the work is being carried out in 3 locations in Ghana, in the Middle Belt in Kumasi, and then far north in Tamale, and then upper west region in Wa. It's a 2x3x2 factorial experiments set out in a split plot design with the treatment randomly distributed in the splits. And then it's been done in 3 replications with 2-meter alley separating the replications, and then 1-meter alley between plots within the replications, plotting the replications. So the main plot here is the cropping seasons, and we have 2 levels of that because we have 2 rainy seasons in the Middle Belt, that is to say Kumasi. The cropping system in Kumasi is different from that in the northern part, and then in the upper west, that is Tamale and Wa. And so for Kumasi, what we are doing is that we have one system to be peanuts in the major season and then maize in the minor season. And the integrity of each plot is maintained to allow the rotation for 3 years. And then the other is growing maize in the major season and then replacing them with peanuts in the minor season for 3 years, also maintaining the integrity of the plot. For the cropping systems in Tamale and Wa, what we're doing is that we have peanut in the first year on a plot, and then the following year is being replaced by maize, and then the 3rd year we go back to peanuts. And then for the second cropping system, we are doing maize on plots, and then it's being followed with maize again in the 2nd year, and then peanuts in the 3rd year. Next slide, please. So for the varieties that we are using, we have 2 varieties per crop. And for the peanut, we have Yenya also, which is an improved variety, and then Chinese, which is a farmer variety. And then for the maize, we have Omankwa, which simply means that which gives life, which is an open pollinated variety. And then Ahofe, which means beautiful, which is a hybrid and orange variety. Next slide, please. And so the subplots, we are looking at 3 levels of input systems. And for the peanut, we have both peanut and maize, actually, we have low inputs, moderate inputs, and high inputs. And all these inputs, the moderate is a buildup of the low input, and then high is a buildup of the moderate input. So for all of them, we are doing good seeds and timely planting. And then for weed management for the low, we are doing just 1 manual weeding, as a lot of farmers do here, sorry. And then for the moderate input, we are doing 2 manual weedings. For the high inputs, we use pre-emergence herbicides and follow them with manual weeding. For disease control in the low, there is no disease control, but for the moderate, we are applying local soap 3 times, and then for the high, we do fungicide treatment for the diseases. Nutrients application, there is no fertilizer application for the groundnut in the low input, but we do NPK application in the initial stages for the moderate input, and then the high input has the NPK applied, and then we also do calcium application to help with pod formation. So for the last part, which is a drying aspect, the resultant pods are then dried on the floor and saved in sacks for the low input. Then for the moderate input, the pods are dried on the tarpaulin and saved in sacks. And then for the high, they are dried on tarpaulin and saved in sealed bags. Next slide, please. A similar thing is done for the maize. However, the only changes that comes in is the insecticide application, which is done for the moderate and high, and that which is done for the moderate, a double of that is done for the high, talking about the number of times that is done. And then for fertilizer application, we do 1-time fertilizer application for the moderate, that is just NPK. And then for the high, we do NPK application as well as urea application as recommended. Next slide, please. So for data, so these are basically the active ingredients and of the treatments that will be applied, and then the time schedules for the application. Please move on to the next slide. So for data analysis will be done with the Statistics 9 software package, and NOVA will be generated and mean separated by the standard error of differences at 5%. The cost-benefit analysis would also be done with Microsoft Excel and then Statistics 9. Next slide, please. So this is just a picture to show how the field is. And so we have peanuts in one side and maize in the other side. And depending on the rotation, maize would follow the groundnut, or the maize would be replaced by peanut, or depending on the rotation. Please, let's move on to the next slide. So I'm going to show you a few of my results. I wish I had enough time to tell you more, but I will just talk about 3 of the preliminary results. And so for this, I'm looking at the effect of the input system and crop on weed population and growth. And here there was a significant interaction for the systems and then the crop. What is happening here is that when we focus on weed biomass, which is to say the weed growth in maize was lower in Kumasi and in Wa as compared to weed growth in the peanuts. And then when you focus on the systems, you would realize also that the weed growth for the high inputs plots and the moderate input plots were mostly lower as compared to the low input plots for both the peanut and the maize. However, when you look at the weed density, that is those on your right, you would see that the weed densities were rather lower in the peanut plots, but higher in the maize plots. And this is an interesting trend to look at, because having a few weeds in the peanuts, but still giving you so much growth, needs to be looked at. Next slide. Okay, so this is talking about the effect of input systems and varieties on peanut disease incidence and yield. And I'll be talking about the yield somewhere else, so I'll just focus on the disease and the home weight here. So looking at, this is just for Kumasi, I did for Tamale and Wa separately. And what is happening here is that the disease, leaf spot disease, both early and late leaf spot, the disease incidence were lower in the high input plots than the moderate input plots and the low input plots. When you look at the severity for the high incidence for both the improved variety and the farmer variety for peanut, you realize that the severity of the disease was very low as compared to the moderate and then the low. However, for the moderate and low inputs, you would see that where the improved variety was cultivated, the severity of the disease was lower, significantly lower than that of the farmer variety. Also, talking about the home weight, which is actually how much biomass the granite is producing and leaving on the plots for the next crop, you would see that the high plots, both the improved variety and the farmer variety produce much biomass, which is to be left on the field for the next crop. A similar thing happened in both Tamale and Wa. Also, for the moderate and low input, you would see that Yenya also, which is the improved variety, produced more biomass, which were significantly higher than that of the farmer variety. Next slide, please. Yeah, let's skip this, and we skip the next as well. Skip to. Skip. Okay, so this is when all the data were pulled together over the locations and then the varieties as well. And so this is just looking at the input systems. The high inputs gave us lower disease incidence as well as severity. And then the home weight, which is actually how much biomass is being produced for them to live on the field for the next crop. The high inputs produce more, which was significantly higher than that of the moderate, and then the low, and then the moderate also produced more than the low. For pod yields, it was seen that the high inputs produced more yields, both pod and kernel yields, but the economic analysis would prove whether it was an economic venture or not. Next slide, please. So this is my work plan, and field work is being carried out in the second quarter and the third quarter of every year from 2019 to 2021. And so this year, I'm almost done with the first season in Kumasi. It's to be harvested this week, and then in about 2 weeks' time, the next for the minor season would continue. Tamale is about 6 weeks old now, the same with Wa, which is also 6 weeks old now. For 2019 to 2021, minor season work will be done in the later part of the third quarter to the fourth quarter. Laboratory analysis for the soil, as well as the seed quality test and other things are also to be done here. And then the data would be entered alongside data collection, and then the writing starts this year. Hopefully, I'm going to submit my work in the second quarter of 2022, defend it in the latter part of the third quarter, and graduate in the fourth quarter. Next slide, please. I would say thank you to the PIs and everybody. Thank you.
All right, thank you very much, Stephen, for the very comprehensive presentation. We've had a couple of questions. Soraya was asking or was commenting that there seemed to be a little difference between disease severity and pod yields, particularly between the farmer variety and the improved variety. Did you expect that? Or do you have any ideas on why that might be?
Okay, so what's happening here is that the improved variety is a bit tolerant to the leaf spot disease, but with the application of the disease control as well as good weed management practices and basically the good agronomic practices, you realize that the yields for the farmer varieties were better because they are well adapted to the local environment. That is what I think, and that is expected. Once you give it the best of treatments, it would outperform one that has been developed for you.
Okay, and another question comes from Helga, similar to one that I was going to ask you about, and that is that these packages obviously involve economics and input and labor costs, and we're just wondering, what are your plans to incorporate those types of information in your analysis?
Yes, so I'm taking data on the cost of everything, inputs, labor, and even time needed for the application of all those treatments and everything that goes into it, planting time, harvesting time, shelling, everything, so that it would be put into the economics to really prove that the system may be better than the other. So they are all being looked at.
Very good. That's good to hear. So I don't see any other questions at this time. So thanks a lot, Stephen, for the presentation, and good luck with the coming season.
All right. Thanks so much.
So we'd like to move now on to our next presenter, and that's Esther Achola. Esther is a PhD student at Makerere University in Uganda. And Esther will be giving us an update on understanding the genetics of resistance to groundnut rosette disease. Over to you, Esther.
Thank you very much. Esther Achola is my name, like it has been said, and this morning I would like to share part of the work that I've been doing with regarding to understanding the genetics of resistance to groundnut rosette disease. So I'm being supervised and guided by Dr. Peter Waswa from Makerere, Dr. Kaleo Kelo from Serere Nasari, Dr. Damaris from ICResearch, and Dr. Josh Clevenger. Next, please. Hello, next, please. Hello. Okay, thank you. On the project that I'm working on, which is the diversity project for Eastern Southern Africa, groundnut rosette disease is really the biggest constraint of groundnuts in sub-Saharan Africa and in Uganda, of course, and the disease results in yield loss of up to 100%, especially when the plant is attacked before flowering. So in the picture there is a typical case of a plant that has been affected by groundnut rosette disease before flowering, that is A, and then B, where the plant managed to survive. And this is actually within the same plot. So the disease is endemic to Africa and its offshore islands, which is Madagascar. And the interesting thing about the disease is it's transmitted by aphids, but caused by a complex of 3 agents. which act synergistically. So it's very interesting to see in the field that we see different symptoms of rosette. We see both green and yellow rosette in the field. And interestingly, if the disease attacks the plant before flowering, most likely we will see more of the green rosette. And then if it's after flowering towards podding and all, then we commonly see yellow rosette, which is something interesting that was observed in the field. So host plant resistance is definitely the most affordable and effective approach because we are dealing with resource poor farmers in Africa, and therefore we just need to make sure that we are trying to provide inputs that are actually cheap and affordable by the farmer out there. And so host plant resistance definitely comes in handy. So, but despite the fact that breeding for the disease has been going on in the region for a while now, it's been evident that there is narrow diversity in the lines or the germplasm that is being used for improving the trait. And also the challenges for a while, little has been reported on the understanding of the genetic control of granate disease resistance. So those are the two areas that that my work is going to try and address broadly in one way or the other, but just to see if we can find other sources of resistance or to be able to understand what is happening with regards to resistance to the disease. Next, please. Yes, thank you. So I'm working with, I'm working with the diversity project, so which involved collection of lines, which were about 1,000 plus, and then they were genotyped, and that genotypic data together with the information from the breeders was used to develop what we would call a co-collection for Africa across the breeding programs, and these were 300 lines. But the seed that came to us had to be increased. And the biggest challenge that I really had again was this disease, that even though we planted even within the screen house and then also planted in the field, the pressure was really high. And even though we tried to spread, it was a challenge. So to be able to do what I'm doing, the main aim here, the general objective really is to try and contribute to granate productivity by improving on the efficiency in selection of resistance in the development of granate disease resistant varieties. So specifically, I'll be looking to evaluate the African co-collection for alternative resistance to GRD and identify genomic regions associated with GRD resistance. Then it will really be important to go on and validate the molecular markers linked to GRD resistance, and then finally develop molecular marker assays and try to see if they can actually be deployed in the breeding program in Uganda and probably in the region. So those are the hypotheses that I'm going to try and test and really see if there's alternative resistance to GRD in the co-collection of Africa, and then also to find if there are genomic regions associated with GRD resistance in the same population, and then try and use specific populations to try and validate the molecular markers that I will identify through the genome-wide association studies in the African collection. And then finally, to really try and test if these markers that we have developed can really be deployed in the program. Next, please. Yes, so for today's presentation, I'm going to talk about objectives 1, some results. And so the work was done this season, which is not yet complete. We just have about 2 weeks or so to harvest. So I planted the African co-collection for phenotyping. So like I shared is we had a little challenge with seed increase. And because of that, I was able to only have about 200 genotypes out of the 300, which could provide enough seed for planting the trials across 2 locations. And I'm hoping that maybe next season I'll be able to add up this number because the seed is getting increased. And so I planted them across 2 locations, which is Serere, where the research institute is, and Nakabango in Jinja, which is closer to the capital city here. And then across 2 replicas. This was done in an alpha lattice design with 20 blocks and 10 plots within the blocks. So I collected data on GRD, and I focused on percentage disease incidence. I collected it at 4 weeks, 8 weeks, 12 weeks after planting. So even I won't collect at harvest as we harvest. So and then for the first objective, which is to identify resistant lines, which might be alternative donors, I will find the means across, I would find the means across the different locations, and then the average mean, and then find those that are actually resistant and try to look at what is happening with the alleles that are responsible for the resistance. And then secondly, I will do a GWAS study. I actually used the data that I collected and then generated blobs in R, which are definitely a better estimate of what could be the true breeding value. And then I used the version 2 of the SNP array to be able to do GWAS. So I did both in analysis. I used both the general linear model and the mixed linear model, which was trying to cater for the kingship. And then I tried to search for the candidate genes on the peanut base to try and see what could be the putative genes on which the SNPs are actually seated on. Next, please. So just to clearly show what I mean by PDI, the percentage disease incidence is just literally the number of plants that are affected or infected over the number of plants that are in that particular plot, and then I multiply by 100. The interesting thing about the disease, it's not like let leaf spot, whereby if it attacks the plot, the whole plot is really affected. With GRD, it can attack half of the plot or one plant. So it just shows you that there is not probably like, let's say, immunity to the disease as such, because you can find plots that actually are able to withstand the pressure, even though one plant may be affected also, but the rest of the plot is all good. So I used that scale, which was by Walira et al. And then, so if in a plot I had 0 to 10% of the plants affected, that I would consider a highly resistant line. Then the ones that are resistant would be 11 to 30, and then 31 to 50 would actually be very susceptible, or susceptible if it is above 50%. So this is a typical example, figure 2 and figure 3. We can clearly see that there's a line there that is highly resistant, and this is one of the lines that was released last year by Dr. Kello here in Uganda, and it's good to see it doing quite well. And then there's another line there that is very susceptible. Actually, by the time the plant, the variety was flowering, the whole plot was actually hit. So I was also able to calculate the area under disease progress curve to be able to see how the disease was progressing over time. Next slide, please. Next slide, please. Okay, my results here for the first objective. So these are box plots to just show how the data really varied across the different stages at which I collected the data. And so at 4 weeks, we couldn't see any serious variation, even though in Nakabango we're already seeing some plants getting hit. And then at 8 weeks, we could see that, yes, the data was more uniform, not uniform, but normally distributed. But we could see that there were more plants that were definitely affected. And then, so at 12 weeks, which is really towards harvest, we could see that even the means were actually closer to 100, meaning that a lot of the plots were actually hit. So we can actually see that at Nakavango, there was quite more variation compared to Serere. And there was also one thing that was important to note. The pressure in Serere was quite high, the disease pressure, but most of the plots seemed to be overly attacked even after flowering, compared to Nakavango, where the pressure started earlier on in the earlier stages of the plant. So there were significant differences for phenotypes at 8 and 12 weeks at both locations. And then at 4 weeks, it wasn't the case. Next slide, please. Yeah, so that scatter plot in figure 6 is just to show where a lot of the lines really belonged. So we could see that of the 200 lines, most of the lines were actually susceptible because they were above 50. So we just had about 31 lines that were only having an average percentage disease incidence of less than 50. And so, well, I just decided to summarize that, and we could see that Uganda had the highest number of lines that were actually resistant, and then second is Malawi. So it's actually interesting to note that, and well, I think a little obvious, because Uganda really has high disease pressure and good screening ability because of the hotspots, and well, the plants are more adapted to the environment. The other place was Malawi, which also I know does some good screening for GRD in the country. Next, please. Yeah, so just to summarize, I tried to look at the lines that were resistant, and they seem to have the same background genetically. And I am just hoping that maybe among the 100 that I did not plant this season, we could find those that probably have a different background of resistance compared to what I actually saw. So that is just a summary. We could see that the best probably 10 lines that performed well actually had the same alleles in the background. And looking at, again, down, which is the worst performing lines, actually, we could actually see that they also had like the opposite allele of what the resistant lines actually had, which is a good thing for development of markers, because now it means that we can clearly distinguish between the resistant and the susceptible. So for this particular objective, I can't clearly say right now if we have found any alternative source of resistance to GRD, since the background, we have many genotypes, but the background does, for resistance to GRD, the background does actually look the same. For now, I'm hoping the results may vary by the end of the work. Next slide, please. So for objective 2, which was to try and identify genomic regions associated with GRD, I was able to do analysis for both 8 and 12 weeks because the data was clearly varying. And this is what I actually found. This is the QQ plot for the values that we used. And so for variation for the data at 8 weeks, we could actually see that there were many SNPs that were significant, but I had to use the significance level of P less than 0.001 because I tried to use the Ben-Ferroni cutoff and I almost did not have any significant SNPs yet at that stage. So all this, when I say 51 SNPs, they are to do with both methods, the general linear model and the mixed linear model. And yeah, they cut across. And then it was interesting to note that Serere had fewer SNPs compared to those that were observed at Nakavango, which were definitely more SNPs. Next slide, please. Okay, so at 8 weeks, I have a summary of the markers and we can see the p-values right there. So it was interesting to note that one of the SNPs was seated on a candidate gene, which is on the fourth chromosome on the A genome, and it was found to actually be some kind of disease resistance protein, which was very interesting to find. Next slide. Yeah, so like I had mentioned, I had also calculated the area under disease progress curve. And so this is just looking at how the data that I used looked like. So you can see the data is either skewed at locations. So this gives us generally a rough idea. It might not really be many genes as such. Of course, now the one for both locations is a combination of the two. But if we look at the individual locations, we can actually see that the skewness probably represents it to be one or a few genes that are responsible for the resistance, just by looking at the distribution of the data right there. Next slide, please. Okay, so interesting data that I found at 8 weeks, because we could also see that this disease pressure had increased and there was more variability clearly, especially for Nakavango. So we had more significant SNPs, and so I could use a more stringent cutoff. And even with using the Bonferroni correction, I was able to identify 29 SNPs that were significant. And one interesting thing to note, looking at two particular chromosomes, the first one, which is on the A genome rather, the fourth one, which is yellow, we could see that there were some SNPs clearly showing that were definitely above the cutoff. And then also on the second one, still on 4, we could see the blue ones having really nicely significant SNPs that were showing. So this is very interesting, because the previous presentation that Dr. Josh presented with the work that they were doing with Dr. Damaris on the population that they were trying to map at ICRISAT actually also hinted that what they were seeing, the significant SNPs, were actually on chromosome 4 on the A. So this is quite good, meaning that it's a good place to start development of markers for this particular trait. Next slide, please. Yeah, so again, we have a number of putative genes that we actually identified. And then again, looking at the one that is in the box, that is again the disease resistance protein that we actually found on the same location. And then it was interesting to find that it was there at 8 weeks, expressed at 8 weeks, and also expressed at 12 weeks, which is a good thing. And then also there were these other 2 SNPs that we actually identified to be on the same gene. But it's interesting to find that a lot of these SNPs that we're able to pick up with the highest significance levels actually are either on A04 or B04, which is really good. And then also looking at the R² values, we can see that these SNPs account for up to 20% of the variation, or they can explain up to 20% of the variation, which is definitely a really good thing to find. Next slide. Yeah, so in conclusion, these results are really promising towards marker-assisted selection work. And then we hope that we shall finish the season by harvesting and collection of post-harvest data and data at harvest, and then also establish the relationship between the disease that we see and the yield and yield-related traits. So I'll be able to do the same and perhaps increase the number of genotypes, and maybe also increase the number of reps depending on the amount of seed that is available for next season across still the 2 locations. Next slide, please. Yeah, so objective 3, which involves validating these markers. The biparental populations have already been developed, but we also have a backup population from Dr. Fonseca that we'll be waiting for, since he has the ability to be able to identify true hybrids at F1, and we'll be able to either, depending on when we hear from him and how, to either plant next season or 2021A. And then for the final objective, this is definitely planned for 2021 because it depends on whatever we find from the work, the first, rather the second and the third objective. Next slide, please. So similar challenges with the rest of my colleagues. The pandemic has really led to transport costs doubling, since we have to social distance even in public transport. And then more online meetings. Stable internet is no longer a luxury. And of course, it therefore has increased costs of what you could have planned to spend on online meetings. And then university has also been closed since the COVID-19 pandemic. As much as we can access our supervisors and all, it has actually affected a lot of things going on, like processing of some of our funds for stipend and stuff like that. So we're hoping it actually gets opened soon. Next slide, please. Yeah, thank you all for listening. And I would like to acknowledge the different partners and definitely thank the Peanut Innovation Lab through the US AID for supporting and funding this project. Thank you.
Thank you very much, Esther, for a very interesting presentation. And it's quite exciting to see that we may be getting close to having a better understanding of both the genetics, but also having a marker that could be used in breeding for rosette disease. Got a couple of comments or questions. One was from Henry Ssendagire on, he was interested in probably discussing with you perhaps separately about the relationship between yellow and green rosette as far as the timing of GRD attack. And he was just wondering, you know, could you point him to some of the literature around that?
So this is just my individual observation in the field. I have not yet seen that really published anywhere. It was just interesting to observe that that is what I was actually seeing on my plots, in especially Makerere.
Okay, good.
Yes, please.
And Isaac was just commented that, you know, in looking, I guess, at some of your hypotheses, you may want to think about them, particularly, I assume, as you develop your thesis, in terms of making sure that the wording is clear and concise to make sure that they're actually testable. Hypothesis. Okay. It's always a challenge with hypotheses, I think.
All right.
And Soraya had a comment just on the fact, I had a kind of a similar comment, you know, that, you know, you've obviously found very highly correlated resistances on A4 and B4, essentially very similar positions. And she was just wondering if you have any explanation for that. You know, is it a genetic effect or a methodological? And similar to that, my question to you was, you know, given that it's such a strong peak, does this give us any indication that there's very little alternative methods of resistance in peanut, that there really does seem to be just a single locus, single gene effect?
Thank you. So I'm not sure what to say about the A and B, but considering that the two genomes are not very different from each other, I would really think around that. But hello? Sorry, I can. So I was saying that considering that the genomes are not very different from each other, the A and the B, I would really think that might be the thing, but I cannot definitely pin it down and say this is just it. But as the work goes on, I could probably be able to find a great explanation for that. And yeah, even before we went into doing this work, talking to a few professors and people around who had done work on granite rosette disease, they definitely suspected that the source of resistance that may have spread across the entire continent could have been from just one particular genotype way back from Nigeria. So they actually said, we suspect that it might just be one source that spread across. Yeah, so, well, that might be proving it, but I do not want to conclude that until I probably finish with the entire population. And then also I was trying to find something in the wild background, even though I have not yet found anything yet, but we might have to try looking. But right now, the evidence seems to point out that it might actually just be, you know, just one or a few genes from one particular background.
Okay, very good. Thank you very much. I think we need to move on, but very good work and very exciting, and we look forward to seeing further progress on it. So thank you very much, Esther.
Thank you very much.
So we'd like to now move on. We're actually going to keep with our groundnut rosette theme. Our next presenter is going to be Henry Ssendagire, who is a master's student also at Makerere University. And he's going to update us on his project looking for the alternative hosts for groundnut rosette disease virus complex. So Henry, I look forward to your presentation.
All right, good morning, everybody. I am Henry Ssendagire, master's student at Makerere University. I am working on identifying the alternative hosts of groundnut rosette disease. I'm working with Professor Mike Diom, Dr. Velma Okaron, and my academic supervisor is Dr. Mildred Ocho, assisted by Dr. Tim. Yeah, next please. It's a beautiful thing to be that I'm flowing in next to Esther, who has done an exhaustive job in her introduction, so mine goes straight to the point. She indicated that the disease is caused by 3 agents, that is the GRV, GRAV, and the satellite RNA. So my role here is very simple. I have to look out for where do these things stay when the groundnut is not in the garden. The groundnut is actually a new crop to sub-Saharan Africa, where the disease actually is. That means that there should have been some crops, that had a pathogen that caused the disease, that moved from those and then came to the groundnut, and then we started having this big problem of GRD. So if I happen to find these alternative hosts, of course, this will add so much to the toolbox of managing the disease. And also in some other cropping systems, literature has it that many have utilized these alternative hosts in management of the crop diseases, mostly the viral diseases. And that gives me hope that as I do this work, I'll have a lot to contribute to this. One of the things that has happened is a research that was again Hello, is all well there?
Yes, Henry, you can continue. We can hear you.
All right, I was having several notifications on my screen. So I was saying that a research done by Dr. Velma Okaron and the team indicated Ssendagire, which is the common sickle pod that is distributed everywhere in the world. And it was positive for the 3 agents of the rosette. Once we have only 2 agents, or 1, because usually the GRV and the satellite RNA will cut together. So that means you can find a plant that has only those. And you can also find a plant that has only the helper virus, the groundnut Rosetta sister virus. So once we have those two happening in different crops, it means those are dead ends. We cannot have any disease to be carried on to the next plants. So for the plant to be as host of this, for the disease to be picked from there, it has to be picked, the three agents must be picked from one plant and then moved to the other plants. And so far, we only have groundnut to be the only natural identified host. Maybe in addition to these preliminary indications from Dr. Carlo Okero and the team. And that is what my research is up to prove whether this is one of, this could be the alternative host, or there are even others in addition to that. Next slide, please. So for me to be able to address the issues, first and foremost, this research of mine is to contribute towards a better understanding of the epidemiological variation in space and time of groundnut rosette disease. And specifically, I have to identify the potential alternative hosts, possibly the natural ones in addition to groundnut of these GRD agents. And in that, since there are some indications, I'll be confirming if Senna obtusfolia, the sickle pod, is actually an alternative host. Then also I would be able to check around to find if there is any variation and diversity among this Apis craxivora, the vector that actually spreads the disease, in the different agroecological zones. And then also for me, if I test, for example, Senna obtusfolia and realize that it has the 3 GRD agents, the two viruses and the satellite RNA, I'll have to do vector transmission studies to confirm that the disease can come from that Senna obtusfolia to the groundnut, then back from the groundnut to the Senna obtusfolia. And those three objectives will give me an achievement to the general objective I have. Next, please. So currently, back to the previous slide. Yes, the current progress so far. Number one, that this work was supposed to start with a survey in which I had to visit the hotspots. Uganda has mainly 3 hotspots. We have the east, 3 hotspots which are mainly in the east, and I was supposed to visit the hotspots and one of the major growing areas to pick out, to get the samples, look around from different weeds showing different symptoms of rosette, and then pick, and maybe even those that were carrying the aphid populations on them, pick those samples and bring them to the lab for analysis. And I did the survey, and I collected up to a total of around 90 samples. And from those, I collected samples from 70 plants, which were showing symptoms characteristic of rosette, of groundnut rosette disease, but had no aphids on them. Then I also samples from 20 plants that did not show any symptoms, but had aphid colonies on them, and specifically Apis craxivora. Then I also picked 20 aphid samples. It was a season where the rains were too heavy, so it was hard for me to find a number of plants that had aphids on them, as many of them were washed off by the rains. As I did the survey and sampling, I collected GPS coordinates of the places where the samples were taken for reference. I took photos of those that were showing the symptoms. In some weeds I found, I wasn't even able to tell what they are, what their names were. So I collected some bits of the plants, like small twigs, and I kept them in a weed album. Those ones I would take to the herbarium for identification in case I need to, depending on what the lab tells me about those weeds. And then also, since I had to do vector transmission studies, I collected seeds. From those plants that were having seeds, I collected them, mostly Senna obtusfolia. I made sure that I go with those. Next. This particular slide is trying to show some of the things that are happening. Like in A, you'll see that That plant has leaves that almost look like groundnut, and that is the Senna obtusfolia plant that I'm talking about. And if you look keenly, you will see that it has some symptoms, areas of chlorosis, yellowing, and I picked samples from that one into that vacuum inner tube, as you can see. Of course, I had to use gloves since I'm dealing with RNA, DNA. Then I put this in a shield. I bought an RNA shield from Zymo, and that was to protect the sample, to protect the integrity of the RNA and the DNA in the sample. Then I transported it in those vacuum tubes you see in C, with ice in a cool box to the lab. As I was doing the sample collection of the from the leaves or from the plants, I also collected some of the sources of what is going to be my source of inoculum. In D, you can see there was a number of such plants, but this I found very interesting. I would find a plant, a particular place where a groundnut has both the green rosette and the yellow rosette. And I found it so interesting and picked, of course, I picked samples from it, and then I uprooted it the way you see in E, and transferred it into those pots, and I took it to the screenhouse, as we shall be seeing as I proceed for the vector transmission studies. Next slide, please. Yeah, also trying to show a bit more of what was happening during the sampling. In G, some of the samples I collected were interesting, and some of them are the ones I'm trying to show here. Like you see in G, that was a plant that was growing. I think that is goatweed. It was growing. It was already showing these rosette symptoms. It didn't really have any yellowing or whatever, but it was stunted, just the same way green rosette attacks a groundnut plant. And the interesting part is that that particular one was growing right next to a green rosetted plant. So if you look at that plant of groundnut in relation to the others, it was having green rosette, and that particular plant was growing next to it. I found it interesting, and yeah, a sample from it was so much worth. Then in H, I found several plants that were having that kind of yellowing. Several of them had, this was just a representation, and I picked samples from that kind of plant also. Then also you can see similar rosette symptoms on this, I think this is a geratum. Yeah, I picked samples from that in I. You see those dots around? I picked samples from there because this looks, again, similar to the green rosette symptoms. I picked samples from there as well. Now, some plants like the one in J had just those, the same way yellow rosette appears in groundnut. When you looked keenly, that plant had, and I picked samples from that also. So that's trying to show some of a picture of where the samples came from. And you know that the quality of the samples is very important in the next steps. Next slide, please. Yes, this one, okay. Then when it came to picking samples, you'll find that like this in J, that is an acacia plant. Now, when you look where the arrow is pointing, you may actually think that is the way the plant looks like. But those are very, very many aphids. If such a huge, I think that was the biggest colony in addition to the one in M. So I would find plants like that. It looks very healthy, no symptoms, but has aphids. And we know that the plant can have the GRD agents and show no symptoms. So I picked samples from that also. Then in K, you'll see that this plant had both the symptoms at the top. You see some sense of mottling, but also the aphid colonies were below the leaf. So I picked a sample from that. Then also in L, at the tip of where the arrow is pointing, at the tip of that plant, I think this is a black nightshade. It also had an aphid colony, and I picked. Now this was, in M was finger millet. It was one of the things that was badly infested with aphids, like you can see. So I picked samples of aphids from all these, picked leaf samples from them, and I took them for the further studies as I'm going to be proceeding. Next slide, please. Next. Yes, and these are georeferences. Of course, I chose the last end up to sample 90. It would show you as I took the sample label onto the virtutina that I used, and then a few details in case I didn't really understand the plant. I would say a weed showing these symptoms, and then of course these are the GPS coordinates as generated by my phone app. I think I used the mobile topographer, and those are the ones I'll be using. Then when you look at this data, these samples collected, I highlighted, I have some that are in red, another one in orange, and then the other one is in black. So that was in order of being essential, because looking at the bit of the inputs I have to use, I may not really do tests on all the 90 samples, but because I was in the field and I observed, there's some that I thought were really, really not a miss if I'm to start with doing my lab tests. So the ones in red are those that I didn't really want. I wanted to start with because there were more sure lines. That is apparently in terms of looking at the symptoms. Then the ones in here, of course, would be the next, and then the others. But you know, this can be surprising. The one you don't actually think would have any would surprise you to be having more. But at least I try to organize my steps in such a way that I know what step to take and when. And then also in these collected samples, many of them were from Senegal, which is folia, because Senegal was almost everywhere. That is the psychopod. It was everywhere that I went to collect samples. So I have, out of these 90, I think I have like 12 samples that were from Senegal. And I really can't wait to see what the lab results talk about that. And many of the samples were, many of the weeds were dominant, like the goat weed was everywhere. Some of them were dominant in all the places, in the 4 places I went sample collecting. And I think that was, yeah, that was also worth noting. Next, please. So in the lab, right now, I am trying to optimize my protocols, right from RNA extraction to the last stages of going to view in the gel. So one of the things that happened that was confusing and actually surprising me, I happened to buy a kit, which was an RNA mini extraction kit from Zymo. And then when I did the first extraction on those first 4 samples, it was amazing. You look at the yield of the RNA and it was really, really low. Then I tried another option that was with the CTAB protocol, and it had better yield. So it got confused me a bit until I had to get guidance from Professor Dioma, and he said, you may see lots of better yield of the RNA with the CTAB, but actually when it comes to the quality, the kit gives more quality RNA. So that gave me more hope and to proceed. I have just one challenge in terms of when it comes to lab work. I don't know if it's because of the COVID, but for some reason, the people who are supplying the lab consumables I have to use have not really delivered yet. They delivered the plastics and a few of the small consumables, but all the other things are still pending delivery. And that is holding my work a bit, but I was able to run a bit of this with a few improvised consumables from the lab. And I also went all the way to doing the PCR, and unfortunately I didn't really have those results ready here, and also viewed was able to view what was on the gel, and at least I have a few lines. So there is so much hope that my protocols are going to work. And I think as I get primers that can do multiplex PCR, I'll be able to run a bit faster. And this bit of the lab is very essential. You see that it's between. I cannot do any other works of the transmission studies until I have all these things in the lab, because I need to be able to know that the aphid I'm dealing with is Apis croxivora. That requires me to do PCR in the lab, and until I do the sequencing. And then these other bits of testing between the transmissions from the source. Is it a clean source? Are the aphids really non-viroferous? Are they viroferous? So this is one thing that an interplay between all the objectives, and it's being in place just really helps me get the work going, and that is what is holding most of my work. But hopefully this week or next week, maybe they'll deliver, and then I'll proceed. Next slide, please. Yes, the other objective 3, like you saw, is about vector transmission studies. As you see that picture below, all those are the cages. These are aphid-proof cages I have set up. I have around 6 non-bioferous colonies running, and I've been able to set up the inoculum sources for green and yellow rosette. And also I was able to start some Senna obtusfolia. And at least the good news was it germinated well, because we had a worry that what if some of these things actually don't germinate. I actually started Senna obtusfolia and a few of those other weeds that I got. So in case I have the lab going, I'll proceed straight away with the vector transmission studies. One thing I noted, at first I had collected samples from Serere, and that is a hotspot. So I was trying, yet I was actually looking out, I wanted to start a non-bioferous colony. So I was trying to question myself, getting a non-bioferous colony from a hotspot, really? That sounded different. So I had to run around in other places that are not hotspots and are not really predominant growers of groundnut, and picked more of the aphid samples. Some I started with 2 aphids to multiply and get stable colonies that are non-bioferous. Next, please. Yes, this is inside the cages. You can see that we know that these the 3 agents are not seed-borne. 3 agents of GRD are not seed-borne. So if you have an aphid-proof cage and you can start your plants clean from seed, that means your plants are free from the GRD agents. So in N, those are some of the groundnuts. I'm using the Red Beauty because it's very susceptible, and also using the Choli White, very susceptible varieties, and the ones I'm using in that I'm going to be using in transmission studies, and also the ones I'm using in culturing the aphids now. Then in O, that is our Senna obtusfolia, picked from different places. I also noticed that the Senna obtusfolia was really was actually different. It was differing even in the places you picked. Would have several of them differing, but I picked from them and studied them, and those have sprouted, ready for the transmission studies. And then in P and Q, that is in those cages we have. I was able to start the sources of the inoculum. In P, we have the green rosette. You see where the arrow on your left is pointing? That one is actually pointing to a green rosetted groundnut. The one I picked, as I had shown in the other first slide, I picked in the pot from Serere, brought to Namlonge where I'm doing the work. And then these are other new plants. So, and then this cage also already has aphids. So the aphids in there to keep, because the other plant may die out and yet you don't have the agents transmitted to the other crops. So right now that is a culture that is ongoing, being sure that the GRD agents are kept on site for the transmission studies. Then in Q, it is the same thing happening, only that this time around it's happening with the yellow rosetted groundnut. And I just can't wait to see how this will happen in the next work. Next. Yes, I would like to thank you very much. I thank the Peanut Innovation Lab and everyone who is really the students, the professors and the doctors for the guidance. I can't wait to see how all this ends up, and I'm really optimistic at seeing what this brings on board. Thank you.
Thank you very much, Henry. Very interesting work and exciting to see that we're maybe making some progress learning the alternative hosts. A couple of quick questions. Helga was asking whether the aphids that you're collecting are all from the same species, or are you going to be determining that with those samples?
Well, basically I'm collecting one type of species, and that is Apis craxivora. So what I'm using as I'm collecting is the knowledge of the visible features that I can look at and tell that this is Apis craxivora. However, we know that even there is variation that exists within the species. And also, so that's one thing I'll be telling apart through the lab work and the analysis and the sequencing that I'll be telling apart to know that if the aphids in Serere, for instance, or the aphids in the hotspot and the major growing area of the groundnut are the same, or they define this way. So that's one thing I'm going to be telling apart. And also to confirm, because now I'm picking with visible signs. So I'll need to confirm, is this really Apis craxivora? And that's one other thing I'll also be doing.
Okay, good. Stephen Arthur was just commenting that he would be interested to talk to you and learn more about the results from your lab analysis on the wheat samples. So, you know, to me, that's one of the purposes of having all of our students present, is that other students can identify interesting work that they'd like to follow up on. So I encourage the two of you to get together after the session today.
Right.
And finally, Jessica was just asking, what are the local uses of senna in Uganda?
Every other time that I happen to move into the field, I realize that senna is actually something that people put into use. One, it's eaten as a vegetable. Several people eat it. There are several names. At the beginning of this study, I knew only one. That was oyado. But as you move to the different regions of this country, especially from where I collected the samples, when you talk, people may not know it until you show them, and they actually relate to it as a vegetable that they actually eat a lot. Then I also noticed that some people use it as a local herb somewhere. And then also I realized that some people use it for I think is it some sort of feed. People cut it down, let it wilt a bit, and then as they are feeding their animals, some actually incorporate it. But this is one thing I was looking at in case we actually confirm that this is one of the alternative hosts. We'd actually go into more studies on this, analyzing possibly what are the nutrient values and what have you, but the best things that can best be put to this crop or any other that happens to be the victim.
Okay, good. Yeah, that'll be interesting information to include in your thesis for all the different weed species, but particularly those who find as alternative hosts for GRD. So I think we'll have to end it up there and move on. We're a little bit behind schedule, but thanks a lot, Henry, for an excellent presentation, and we look forward to hearing more about it, and good luck.
Thank you.
So I'd like to move on now to our next speaker, Leslie Commey, who is a student at Texas Tech University. So one of our few students who was able to do his studies in the US. And Leslie's going to update us on his work to develop an A. flavus resistant peanut using seed coat biochemical markers. So over to you, Leslie.
Thank you very much, David. Good morning, everyone. And today I'll be talking about developing Aspergillus flavus resistance using seed coat biochemical marker. And the main PI for this project is Mendu Venugopal. Please next. Our collaborators are Dr. Magburu of Texas A&M AgriLife, then Hamidou Fallu from Ikrisat Niger, and Hikrishan Sudin from Ikrisat India. Sorry. Next. So first of all, we know that A. flavus causes aflatoxin contamination, and aflatoxin has been a major problem for the past 50 years, with people approaching it in various sense, with some of them approaching it from the molecular perspective, using molecular techniques to find or to be able to produce A. flavus resistant peanut germplasm. Others have also looked at harvest management techniques such as using aflasives and also drying of the peanuts and other factors. However, our project focused mainly on the post-harvest condition. And what we did was, peanut seed coats are usually the only outer layer that are present in the peanut after the shelling, and that is where that's what is the only outer layer that is used during processing. So what we did was we tried to use, there are 3 different types of mechanisms that are used to identify resistant A flavors germplasm lines. So one of them is using in vitro seed colonization. Another one is using pre-harvest aflatoxin contamination. And also the other one looks at aflatoxin production. So what we did was we deployed the in vitro seed colonization to find out resistant germplasms and susceptible germplasm. What's the difference? So what we did was we introduced the pathogen about 1 × 10⁻⁷ onto the seed coat of sterilized TMV2 and 55437. And after doing that, we realized that for 7 days, we realized that 55437 did not produce much fungus as compared to the TMV2. So based on that, next please. We hypothesized that because the outer layer, the seed coat is the only outer layer protecting the peanut as at the time of inoculation with the A flavors pathogen, then it could contain some biochemicals which could mediate A flavors resistance in peanuts and will provide a quick and efficient method to reduce aflatoxin contamination. Next. Based on that, we hypothesized these 3 objectives that we use these 3 objectives to see whether we can be able to meet our hypothesis. So first of all, what we did was to identify specific biochemical responsible for A flavors resistance in seed coat and use it to develop a biochemical marker. This was to just, because we know that there are some components in the seed coat, either the biochemicals or the structure, which could be attributing to the resistance found in 55437. We decided to dive into the phenolic compound since it has been reported in literatures that they have some antifungal potentials. Then our second objective was to identify seed coat mediated A flavors resistance in field and also using in vitro seed colonization test. This is done in the sense that we are going to look at the different seed coat colors, which primarily we have the purple, the red, and the tan color, which are the main seed coat colors in peanuts. Then our third one is to develop an A flavors resistance line for field development in target country. So this is just a cross between TMV2 and 55437 and see whether we can be able to develop or introduce the resistance from 55437 to TMV2. Next, please. In order to start this work, what we did was we isolated the A flavor strain from an old peanut stock about 6 years old, which we got from Dr. Bureau. As from this picture, we can see that there are 2 Aspergillus species on it. We have Aspergillus flavus, which is the green, and we have the dark one, which is Aspergillus niger. What we did was to obtain a pure culture, so we subcultured it and allowed it to grow on a potato dextrose agar media. Next, please. After that, what we did was we have now phenotypically characterized that this is A flavors, but we need to have some molecular proofs to show that the strain that we've isolated is purely A flavors. So what we did was we used PCR techniques and we know that the Aspergillus flavus, the gene has a band size of about 538. So after running 5 of our isolates, we found out that 4 of them was able to contains the A flavors gene and shows that they are clearly containing A Aspergillus flavus in it, the fungus in it. Next, please. So aside that one, to also go and see whether the aflatoxin, the A flavors strain that we've obtained contains is aflatoxinogenic. We are trying to see, we use two methods. This is normally a method used in Africa and other developing countries, since they don't have like the HPLCs and stuff to be able to detect A flavors. So this one is, the first one is beta cyclodextrin test, which requires just using 0.01% of beta cyclodextrin and viewing it in a UV lamp with a wavelength of about 365. Clearly, if there is fluorescence, it shows that the A flavor strain you have contains aflatoxins or is capable of producing aflatoxin. Another one is to use the ammonium hydroxide test. With the ammonium hydroxide test, what you do is you expose ammonium hydroxide vapors to the A flavor strain. If you produce the plump red color, it shows that the A flavor strain you have contains or has the characteristics of aflatoxin in it. And this is the control, which was exposed to ammonium hydroxide, and it did not show any plump red color. Next. Also, what we did was we tried to use molecular characterization to also see whether apart from the biochemical characterization we've done, does the strain still contains the aflatoxin gene. So from this picture, we could see that it contains about 4 of the aflatoxin genes, which are required for A flavors, aflatoxin to occur. Next, please. So based on this strain, what we did was, after we have identified the strain and also we have been able to state that the aflatoxin strain, the A flavor strain we have contains aflatoxin, what we did was we quantify it using hemocytometer. And based on that, to tackle our first objective, which is to identify specific phenolics, we first decided to prove our hypothesis. Next, please. So we proved our hypothesis by extracting seed coat phenols from 55437 and TMV2. And we realized that seed coat extract was extracted using acetone, acetic acid, and water. And we did it overnight. And the extract was then dried out using rotary evaporator. And then 1 ml of the solution was made using distilled water, and it was introduced into a PDA media. It was mixed with a PDA media and poured on the plate. After that, A flavors strain was placed in the middle of it, and radial growth assay was taken for the 8 days. After 8 days, we realized that radial growth assay for the control was complete, and TMV2 radial growth assay was a bit bigger than 55437. This clearly shows that there are some compounds in 55437, which is the resistance lines, which is mediating A flavors growth more as compared to TMV2 and control. This experiment was done for 3 independent replications. Next one. After 3 replications, what we found out was that we took the data for 8 days. So we calculated the average radial growth for each and every replication. And we realized that among all the 3, the average radial growth for 55437, which is the resistance line, was about 2.7, and that of TMV2 was about 3.4, with the control being about 4.5. And it was significantly among all the 3. Next, please. So then we decided that, okay, now we've been able to find out that the biochemical is present, and it's in the peanut seed coat, and it's inhibiting A flavors. So why don't we go a step further to try and find out, let's say, which biochemical constituent is responsible for the resistance we are seeing in A flavors. Actually, for this one, it's ongoing now, and the update on it is that we've been able to get some biochemicals, and we are also testing using radial growth assay analysis to see whether which one corresponds to and in which combinations are these biochemicals interacting to mediate A flavors resistance, which will come later, maybe in our next presentation. Next slide. Another objective was to use in vitro seed colonization assay to try and find out if we can be able to identify other resistance lines. So what we did was, because 55437 had a tan color, we requested different forms of seed coat color in an attempt to find out whether we can use just basically phenotypic seed coat color to differentiate between resistant germplasm lines and what they have in common, or get a subset of resistant germplasm lines which have a particular color, so that breeding programs will then look out for, let's say, if they want to introduce it into their breeding program, they are going to look out for only seed coat colors of this color in terms of aflatoxin breeding. Next. So from there, we got 11 lines from USDA, and we grew it in our field, and we tested them for A flavors resistance using in vitro seed colonization. This picture depicts the various seed coat color. We can see tan, we have some which is variegated, we have some which is purple, we have some which is red, we have some which is white, which is Tephras 13. And we have some which are both colors. Interestingly, when I dived more into the assertion, I found out that what those who collected the seeds from their center of origins took some from the marketplace. So some of the lines were mixed plots where they have some variegated color of dark purple and reddish color. Next one, please. So after doing in vitro seed colonization, which basically is just introduce or sterilize your seeds using 20% hypochlorate, then introduce your pathogen to it, then look at the number of infestations that take place for a period of 8 days, since our radial growth assay was full completely 8 days. So we found out that after 8 days, we had PI such as PI 544346 showing greater resistance as compared to even the known resistance, which is 55437. And then we had subsequent ones which were in par or a little bit lower than 55437, but then we took them into consideration to see whether maybe they could also help us in understanding which biochemical is responsible for A flavors resistance. Next slide. This is just a graphical representation of the lines that we screened, and you can see clearly that 544346 had a greater resistance as compared to the control, and the others such as PI 54350 showed a little bit, some were a little bit higher and some were a little bit lower than the 55437. So these 4 lines were what we selected, and we grew them under drought conditions. Next slide, please. That is, we are growing them currently under drought conditions to try and see whether, one, what is the aflatoxin contamination or the A flavors contamination level after growing it in drought conditions. And also we are going to see whether the phenolic compounds that we obtained using HPLC, the specific phenolic compounds, whether there is compositional changes as a result of drought conditions or not. And also we are also going to screen other USDA lines. So based on the data we have obtained, we ordered about 56 lines from USDA, which has basically 2 types of seed coat color. One was the tan color, one was pink tan, and we took some mixed plots to see whether we can be able to identify resistance lines among the mixed plot seed coat color types. Next slide, please. So this shows the our lines that we grew under drought conditions, which is currently ongoing, and we are looking forward to taking some data with this in terms of the number of pods formed. And also, if possible, we are trying to see in two ways, whether also the phenolic compounds, apart from the phenolic composition changes, whether maybe the A flavor strain, because we know it affects peanuts about 3 to 4 weeks before planting season ends, we isolate maybe the seed coat we take the seed coat from the germplasm at that particular period and see whether those strains have already been infected already with A flavors, and we introducing the pathogen just increase the tendencies of the A flavors infestation occurring during in vitro seed colonization or not. Next slide, please. Next slide, please. Also, we obtained a cross from F5 lines from Dr. McBureau, which is a cross between 55437 and Schubert lines, which is an high oleic variety. And here, what we specifically wanted to do was to determine whether those lines that were produced contain any line was responsible for resistance, or any line contain any resistance in terms of A flavors from the lines that Dr. Bureau has already produced. And among these lines, what we found out, this please. We found out that none of them showed greater resistance as compared to 55437. And we are hoping to see whether to understand what attributed to that, because it's a cross between a highly resistant line and Schubert, and we were expecting to see some resistance in any of the lines. I'm still talking to Dr. Bureau on it, on what we want to do with this. Next slide, please. Thirdly, our third objective was to develop A flavors resistant line from TMV2 and 55437. So what we did was we did reciprocal crosses, and we got few seeds, about 6 or 7, and we generate F2 during the winter in our greenhouse. And currently, because of the COVID situation, we only grew the lines because we couldn't have time to get access to the time when the planting season started. So we are currently in the F3 stage, where the seeds have already been planted, and we are going to look for putative hybrids using the leaves instead of the seed, which is an alternative, but ideally should have been the seeds, to determine whether our crosses, we have any hybrids between them. And possibly after that, what we want to do is, after we grow our F3 seeds on the field, we will screen them for A flavors resistance using in vitro seed colonization. And possibly, if the biochemical marker has been developed, or we've been able to identify the specific phenolics, we try to develop a biochemical marker and use it as an alternative for the in vitro seed colonization, and compare to see whether the two of them are in correlation, and possibly grow the F4 lines. Next, please. So conclusively, we could see that our radial growth bioassay data indicates that seed coat biochemicals could be a reason why A flavors growth was reduced in 55437 as compared to TMV2. And we also find out that PI 544346 and PI 544350 showed a little bit resistance more than the already known 55437, and could help facilitate or could help us know which specific phenolic is responsible for A flavors resistance. Next slide. I want to thank everyone for listening, and also Peanut Innovation Lab for giving me this opportunity to do this study, and also Texas Tech as well. And next slide, please. Yeah, I'm done for that.
All right, thanks a lot, Leslie. Very interesting work. I've had a few comments or questions. Helga was interested in kind of knowing what you might be doing and thinking about the potential impact for adoption of lines based on their seed coat and the effects that it might have on taste and cooking qualities. And I think related to that, Maria Belota just mentioned that, you know, if you're interested to follow up, say, on blanchability or anything of these lines, she'd be more than happy to talk to you about how she could help you get that accomplished. I don't know whether you have any.
Okay, so with the blanchability, I think I'll contact her to see more on that. And also the removal of the seed coat is just to try and understand, we are removing the seed coat and we are extracting phenols from it to try and understand what is causing resistance in 55437, which is a resistant line. So the seed coat is just necessary to explain resistance or susceptibility, but it doesn't have anything in relation to improving like the seed coat color or something, because I feel like as for that work, that is not what we are doing. Ours is just to try and understand what is causing disease flavors resistance in peanuts, and if possible, use that mechanism to develop a rapid technique that is going to be used in developing countries, since they are not able to detect whether the germplasm lines or the production lines that they've produced contain aflatoxin or not. And ELISA tests and stuff are very expensive. So we are trying to come up with a farmer-friendly technique, which is going to be simpler to determine A flavors resistance, so that they can be able to reduce the number of bans that goes on in terms of exporting peanut material from Africa to Europe and America.
All right. I think another question kind of related is, you know, probably aware that, you know, just reduction of growth and the presence of fungal growth doesn't necessarily correlate all the time with the level of aflatoxin contamination. And so I'm assuming that at some point you'll be looking at that in terms of whether or not lines that actually inhibit fungal growth also reduce the level of aflatoxin in the seed.
Yes, that's with one of our collaborators. So what we want to do is we are going to send the lines to, because in US currently, aflatoxin, you need certain approvals. We started applying, but the process has been halted because of the COVID situation. But however, we are going to send it to Equisat Niger with the idea that they are going to screen for aflatoxin, because it's less like the protocols needed for you to do aflatoxin test is less stringent over there. So we are going to send the lines that we have seen that they are A flavors resistance to that side to see whether those lines correlate in terms of aflatoxin level, whether there is a correlation between the lines that we have identified to be A flavors resistance correlate in terms of aflatoxin contamination. I hope I was able to answer the question.
Okay, yeah, no, it's clear that that's a very important component to the study for sure. Okay, so I think we'll have to move on, but thanks a lot, Leslie, for a very interesting presentation, and we look forward to hearing progress and in the work. So thanks a lot.
Thank you too.
So I'd like to move on to our next speaker, which is Ahmed Seidu, who is a PhD student at the University of Development Studies in Tamale, Ghana. And Ahmed's going to be speaking to us about improved peanut production and pest management for enhanced yield and quality.
Okay, thank you very much. Good afternoon and good morning to everyone. Like it's rightly said, my name is Ahmed Seidu with CSR SARI, that is Savannah Agricultural Research Institute. I'm doing a PhD with UGS University for Development Studies. and I'm going to present on improved peanut production and pest management for enhanced yield and quality. Next. Next, please. My supervisory team comprises of Prof. Israel Meku, who is with the UDS, Dr. Mumuni Abdullahi with SARI, and Prof. David Jordan with Northern Carolina State University, US. Next. Peanut is an important crop in the whole world, of which Ghana is not exempt, particularly the northern part of Ghana, where the production of peanut is widely coming from. It is an important source of food and feed, and is also consumed as peanut butter, peanut oil, confectionery snack, roasted, and also used in soups. Next, please. Peanut production is also improves the fertility status of soil and serves as an excellent component in crop rotation. However, its production is constrained by pests and diseases, including aflatoxin contamination. Again, farmers do not also apply the required inputs, especially fertilizer, to boost their production. Next, please. This therefore results in low yields, sometimes as low as 400 kg or less in less developed countries like Ghana, compared to about 3,000 kg or more elsewhere, that is the developed countries such as US. Moreover, aflatoxin levels is often high in produces, threatening human health, as well as limiting marketability internationally. Next, please. This therefore leads to the design of these experiments to, that is, determine the main effects, that is, of improved production and pest management on peanut yield and quality. Next, please. The specific objectives of this experiment is to determine the effect of planting dates at different geographical locations, the effect of variety on yield and aflatoxin contamination, the effect of fertilizer application on yield and aflatoxin contamination in peanut. Next, please. Also, to determine the effect of improved pest management on yield and aflatoxin contamination, the effect of improved pest management and drying as a production package on contamination, and a combination of the above mentioned factors, that is variety, fertilizer, and production package on yield and aflatoxin contamination in peanuts. Next, please. The experimental site, 3 locations were used, that is Nyankpala, Wa, and Kumasi. And the research force of institutes of the CSIR, that is SARI, and crop research were used. Next, please. The design used for this experiment was split treatment arrangement in a randomized complete plot design with variety as the main plot at 2 levels, that is Chinese and the SARI Not 2, and fertilizer as the subplot, that is fertilizer applied and no fertilizer applied plots, with production package as the subplot, that is the improved management practices as against farmer practice. Next, please. With these 3 factors, with their level combinations, I came out with these experimental treatments. That is 8 experimental treatments in all. Next, please. SARI selected experimental sites for the 2 locations up north, and the crop research did for the south. Plots at each site were plowed and harrowed for before planting. And the plots were planted on flats according to the experimental design used at 3 different planting dates across the 3 locations. The planting dates were mid-June, and mid-July. Next, please. Considering the number of treatments with the replications and the planting date, 96 observations are considered. And with these 96 observations across the 3 locations for over 2 years, 576 observations will be encountered. A plot size of 5 meters square were used, with 1 meter alley between plots and 2 meter alleys between factors and replications, respectively. Next, please. This was the experimental layout, full layout, with the 8 treatments that is considering for comprising of a full replication, and these treatments were randomized in the subsequent replications and the planting dates as well. Next. Data collection comprises, but will not be limited to cropping history of the fields, baseline and post-harvest soil physical-chemical properties of the field, daily rainfall, minimum and maximum temperature, as well as humidity, also be collected throughout the season. Next, please. Associated insect pest, example, thrips, aphids, and soil arthropods, and their populations all be documented. Insect pests will not only be limited to the thrips and aphids, which are foliar insects, but other insects that will be encountered on the field will also be taken note of. Data will be collected on them, and the soil arthropods too. We have white grubs, we have millipedes, and we have the wireworms, which are the major soil arthropods. Those are the insects data will be collected on. Disease spores, leaves, and early and late leaf spores, as well as rosette on a Florida scale of 1 to 10 will be used. Associated with species and density before weeding will also be collected. Next, please. Number of pores per stand, percentage damage caused as a result of soil arthropod damage, number of bags of pox produced for each field, and pod yield, that is in kilograms per hectare, and percentage shelled out calculated for each treatment will all be documented. Next, please. Aflatoxin contamination after drying, but before storage. Physical quality of peanuts, that is sorting out and percentage root kernels, as well as economic data, all be documented. Next, please. This is my activity timeline. The first aspect, that is land preparation and experimental setup, is done. Both monitoring and data collection is ongoing. Now we have taken soil physical-chemical properties, that is before planting, and it is the soil department for them to analyze. The sample will be taken after harvest, and we have started collecting data on the insect pest, that is the foliage insects, and the soil arthropods done at harvest. And data compilation and screening will start next month, and data analysis, data processing and analysis will be done later. Analysis of data will be done depending on data collected. The agronomic and the protection data will be analyzed using SAS or GenStat software 12 edition 12. The economic analysis will be done using the economic softwares, and the aflatoxin contamination after samples taken will be taken to the lab for the analysis. Yes, please. Challenges. Erratic rainfall at the beginning of the season affected the planting, and the trial sites are wide apart and involve lots of travel for field activities from Nyampala to Wa and from Wa to Kumasi and back to Nyampala. A lot of traveling, and it's a constraint. Next, please. I would like to thank Feed the Future USA and all the partners involved, not forgetting all the listeners. Thank you very much for your attention.
All right, thanks a lot, Ahmed. Very good presentation. I think there was just one general comment from Isaac Mende that you might want to think about as you, particularly as you begin to develop your thesis, to make sure that your objectives are clear and build on what's already been done. I know there's been a lot of work, as you mentioned, in this area of looking at production packages. So, clearly indicate what is new in the work that you're doing and why it will build on that previous efforts.
Okay. Thank you very much.
I think one comment that I had, it was made, I think, to a previous student as well. Are you going to be able to look at the economic and labor input aspects to these different packages as well?
Come again?
I was just wondering whether you're going to be able to look at the economic and labor implications for these different packages.
Oh yes, data is being collected on all the activities that is going to be cost involving, and this one will be considered for the economic analysis later. So it will be considered.
Okay.
Thank you very much.
Good. All right. Well, thanks a lot, Leslie, and good luck with the rest of this season. A lot of data, a lot of plots to analyze.
Mm-hmm.
Wish you all the best.
Thank you very much.
So I'd like to now turn to our last speaker for this morning, and that's Tabitha Lomotey. Tabitha is a master's student at Makerere University in Uganda, and she's going to be presenting on the genetic studies in groundnut for resistance to rust disease.
Hello. Good morning, everyone. My name is Tabitha Lomotey, and I'm presenting on the genetic studies in groundnut for resistance to rust disease. And my supervisors are Dr. Thomas Odong, Dr. James Mololo, and Dr. David Kalli. Next, please. Next slide. Okay, so rust is caused by Yersinia rachidis, and the picture on the left is showing the pistils, which look like orange rashes at the underside of the leaves. And this disease alone can cause up to 50% yield losses, as reported in some areas in India. So in Uganda, the disease is irregular and localized in the mid-altitude areas like Serere. But with climate change, the rust is likely to become a big problem in Uganda, either through greater frequency of tropical storms that move inoculum from the southern part of Africa, or extending the range over which the pathogen can overwinter. Therefore, ICRSAT Malawi and Nasari have developed some resistant lines. However, we do not know, we've not evaluated yet for some key traits. And then there's also limited information on the rust resistance and the lines at different locations. As well, the mode of inheritance of rust disease using Q parentals has not been clearly understood. Next slide, please. So for my general objective is to contribute to knowledge in the development of rust-resistant varieties suited to Uganda, specifically to evaluate morphological, agronomic, and yield-related traits in high altitudes, to determine the response of granite lines to rust at high and low altitude, and then to also determine the mode of inheritance of rust disease in granite lines. I hypothesized that granite lines show variation in agromorphological traits due to differences in the locations. And secondly, rust severity vary at the different altitudes, with high altitude showing higher severity. And then for the inheritance, I hypothesized that inheritance for rust resistance in granite is non-additive. Next slide, please. Okay, so I'd like to give some preliminary results so far on what I have done. So this is a combined analysis of variants that I've done. I have 2 replications, 20 genotypes, and this was done in 3 locations. So there's significant difference in the 3 locations as seen on the table. Next slide, please. Okay, so for days to flowering, we can see that there's not much variation in Kabale, and in Bukinyanya, the highest days for first flowering was on the 72nd day, and in Serere, 31 days after. Well, this I think was due to the variation, the temperature variations and the altitudes. When we look below, we see that Bukinyanya has a temperature of 20 degrees, Kabale 19 degrees, and Serere 24 degrees. Kabale, the temperature over there is very, very low. So I think that was what caused the delay in flowering. But then temperatures in Serere are relatively higher, so it's helped a bit. Next slide, please. Next slide, yeah. Now, days to 50% flowering. Well, I think the same thing with the temperatures, which caused a delay for Kabale, like a 2 weeks difference for 50%, and there's no like variation in it. And also for Bukinyanya, it took like a week, and in Serere as well, a week to attain 50%, and the days to 50% flowering. So yeah, let's move please next. Okay, so for plant height, Serere did better as compared to Kabale and Bukinyanya. Kabale did very poorly because of the temperatures over there. As a matter of fact, the plants were very, very short, like as though they were hit by reset, but it was just because of the temperature, and some kind of suffered from like chilling effect. You could see that the roots were poorly developed. And in Bukinyanya, well, they were fair, not as Serere, but it was able to come up though it had a lower temperature, but then the altitude was better than Serere. Next slide, please. Okay, so with the temperatures in Kabale, it prevented yields. So there was no yield for Kabale. But then in Budianya, the yields recorded in Budianya was way higher than the ones we had in Serere. Well, the temperatures were low, but then I think maybe physiologically, when temperatures are lower, it enables more yields to come out because it gives more time for maturity. But then in Serere, because temperatures are a bit higher, kind of like quickened maturity. So Budianya gave better yield as compared to Serere. Please, next slide. Okay, so this is my principal component analysis, and here PC1 and PC2 gave a cumulative of 69.430, of which PC1 gave a variability of 43.206, and realized that in PC1, plant heights did very well, which influenced the variations we can see between the genotypes. And in PC2, 50 days to 50% flowering also influenced the variability in the genotypes. So we can see that in the figure on the left with the angles showing that pod yield and plant height are positively correlated, and then 50 days to 50% flowering is negatively correlated with the plant height, and also days to flowering is negatively correlated with the pod yield and the plant height. Next slide, please. Next slide, please. Okay, so with the correlations, as you can see on the table, days to first flowering and days to 50% flowering, there was a correlation which is positively and very highly significant. Plant height and days to 50% was negatively correlated, but was still significant, highly significant. But then when we look at pod yield and days to flowering, there was no significance. Yes, basically. Kindly, next slide, please. Next slide. Okay, so this is to show how the genotypes fared in the locations. So with the Amy, realized that the genotypes, okay, so from the previous slides, I have listed my genotypes. So genotypes, genotype 10 and 12, and then 16 did well in Buginyanya, and then genotype 5 and then 19 did well in Serere. And then also when we come to the GGE on the right, we realize that genotype 13, 12, and 10 did very well. Like it's ideal for, I think, all the environment. I think both Buginyanya and Serere. Yeah. Next slide, please. So for my objective 2 is to determine the response of groundnut lines to rust at low and high altitudes. So in Serere, there was some symptoms of rust at 115 days. It took a longer time for us to show, but it was able to show. However, in Kabale and Buginyanya, I think because of the temperature ranges, rust was not able to show. So there was no rust data taken in these environments, only for Serere. Next slide, please. So as a way forward, because Kabale and Serere, Kabale and Buginyanya did not give any rust. So therefore, an experiment in the screen house would help so that I'd be able to inoculate and then compare the data with what I have in Serere, on the field in Serere, to be able to monitor the rust. And also for the inheritance study, which is my objective 3, because of the pandemic, well, it's ongoing in Malawi, but then I couldn't get a chance to travel. And then I'm also embarking on this new objective to determine the genetic variation of the pathogen across farmer fields in Uganda and then in the southern part of Africa. So I've been able to do the optimization, was able to isolate the DNA for this objective. So for now, I'm at this stage and I'm hoping to now travel out and to other farmers' fields in Uganda to collect rust samples for this objective. Next slide, please. So as a take home, I'd like to say that all the genotypes in Buginyanya had higher board as compared to Serere. And the conditions in Kabale did not favor the growth and production of the genotypes under study, including rust. So I recommend that Kabale should therefore be a site for longer duration specific genotypes. I'd like to acknowledge these bodies for the help and all the support they've given me. Thank you so much, Peanut Innovation Lab. Thank you.
Thanks very much, Tabitha, for a very good presentation. We've had a couple of comments, and I think Maria Bolota commented that, you mentioned that these different sites have different average temperatures, but it might be better to actually use a growing degree day from planting to maturity as a better measure of those differences. So if you have any questions, you need to follow up on that.
Okay.
Soraya was interested, clearly there's pod yield differences, particularly between Serere and Buginyanya.
Yeah.
But she was wondering whether you think there might be anything else other than simply a temperature difference there.
Okay. Okay.
Something to think about than just, maybe other factors as well, diseases, other stresses.
Okay, so I would also like to do some soil analysis, also to check maybe the soil samples in Buginyanya might have some characteristics which are maybe better than the ones in Serere. So I'll consider doing that to find out that.
Maria also mentioned that there's a relatively simple crop stress index that you might want to use to show differences in terms of heat and precipitation. So she's given a reference, but you may want to contact her and get more information that might be useful as well. Soraya was interested in what method you're going to be using to determine the genetic variation in the rust.
Yes. Okay, so for the lab work, I'll be using some markers to check for the regions where the rust is virulent. So I have some primers, some of the ITS, and also some to check for the cytoplasm. Yeah, so that is what I have ordered for so far.
Okay, I think Soraya may be in contact with you to talk a little bit more about that. So make sure you're using the best technology. Okay, good.
Okay.
Just trying to look through if there was anything else. I think there was a comment from Isaac Mende as well, that just to make sure that when you're doing your rust assessments, you're basically analyzing them at relatively the same level of maturity of the crop, so the same number of days at all sites. Yes, at some point it wasn't clear whether that's the case, but that would be very important. I mean, it always is in any kind of disease ratings, to make sure that you're comparing them at relatively the same stage.
Okay, I'll check that.
Okay. I think that's all the questions, unless anyone else has anything more to ask. Okay. Very good. Well, thank you very much, Tabitha, and we look forward to hearing of your progress, and all the best for a good year. Thank you.